Thanks very much. Welcome to UC Berkeley. For people that haven't been here before, I think you'll find it's quite a nice campus. We are running a little bit, so I'm going to try to speed things up just a little bit. If you go out the street here and you go all the way down the end, this is the Innovative Genomics Institute. There's a building there. It used to be the Energy and Bioscience Institute that BP basically built for Chris Somerville and company. And then 5 years ago, Jennifer Doudna took over this institute. Just a little introduction to the IGI. The IGI is just not agriculture. It has like 4 components to it. Biomedicine and human health is a major component. Sustainable agriculture is also advanced genome engineering, Jill Banfield, discovery of new CRISPR systems, and so on and so forth. But also we feel it's very important that we have to have a public impact component where we try to educate the public about gene editing so we don't make the same mistakes that we did with GMOs, okay? So you can go onto the website there and you can see a lot more what's going on there. So obviously sustainable agriculture is essential for the future. We're basically going to have to increase food production by a tremendous amount on the same amount of arable land, but how are we going to do it in a net zero farm situation? So we have lots of projects that I'm a part of some of them, but not all of them, and I'm not going to talk about all these, but just to give you an idea, I'm going to talk about genome-edited plants for disease resistance to reduce pesticides. We're using gene stacking and durable resistance genes using CRISPR-targeted insertions, very similar technology. We collaborate with Corteva in this particular case. This is, I'm not going to talk about that either. We also have developed very robust RNP editing with HDR, wheat, rice, and several other plants. And again, with a collaboration with Corteva, we also find that baby boom bushel is essential for increased HDR events in monocots for sure. We also work on cassava, where a group has actually moved the cyanide genes from cassava. I'm not going to talk about this. We're interested in improving water use efficiency. I'll give a little boost today for Nicholas Carabolius, a graduate student who will be talking about this later on today, who's developed a very nice system for actually manipulating the stomatal density on plants to give water use efficiency. We're also embarking on systems. We're altering root systems to improve water use efficiency. And then also we're interested in reducing fertilizer dependency by gene editing various things for transporters. And we're not going to talk about that. What I'm going to talk about is, well, the other thing is that, again, when I first came to the IGI, which is almost close to 6 years ago, Jennifer Doudna asked if I would, be the director, and I said sure. She had a lot of philanthropic money, so that was quite nice. And so she said, "What are you going to do first?" I said, "Well, the first thing you have to do is you have to set up a robust plant transformation and editing facility." Okay. Most academic institutes don't have such things. Companies like Corteva and Bayer and so on and so forth have these types of things. And so what did I do? I went out and recruited Myung-Jae Cho, who actually worked at Corteva. He also at Berkeley, but Myung-Jae was also involved with Bill Gordon-Kim in developing a lot of the baby boom bushel type of technology. So MJ is transformed over, we call him MJ, the Michael Jordan of tissue culture. Essentially, he is able to transform and regenerate 30 different crop species, and he's a very talented individual. So we have a very robust group that we've set up. And I'm not going to go in great detail here, but we're able to do wheat transformation both in agro and we can do it in lines that other people can't do it because of MJ's technology. Rice transformation, a little bit easier. We work on for wheat, rice. These are the crops that feed the world. The other thing, we made a decision, since a lot of money at the IGI, it comes from philanthropic sources, we decided to work on crops that feed the world for developing countries, okay? So that's a theme here also. Also tomato. We also have work, as I told you, in cassava. We have that very robust transformation there. And also, probably the greatest feat that MJ would say that he did, can you not? Okay, cool. That we have now developed an extremely robust cacao transformation. And MJ will tell you this is the most recalcitrant plant system there is. And he's been able to do this now, and we got a lot of projects. This is work is funded from gift money from Mars Corporation. So they're interested in various disease traits and other traits that we're currently editing in cacao. So here's your green chocolate. So I'm going to talk about several examples now of targets for disease resistance. This is going to be gene editing 1.0. It's the easiest thing to do, is to identify single genes and to make gene knockouts in particular genes. Okay. For many years, I've been working on the disease of pepper and tomato, called bacterial spot disease of pepper and tomato. And it's caused by 5 different species of Xanthomonas. And you can see what the lesions look like. And it's a serious problem in many parts of the world where you have a high humidity and warm temperatures, and oftentimes it can actually destroy the entire crop. The other problem is that there are really lack of disease resistance genes that have been identified in wild species of either tomato or pepper that have lasted over a period of time. And the way they try to control this disease is actually by spraying copper that's with a mancozeb, which is a fungicide, which facilitates the copper uptake into the thing. But all those compounds are pretty toxic. If you go to packing sheds in Florida, you'll actually see it contaminated with copper. Copper is a pretty nasty compound, believe it or not. They use it in organic agriculture, so watch out for your organic food. So anyway, important disease. So our approach here to construct durable resistance for these in any crop is we use CRISPR-Cas9 to construct precise mutant alleles that confer resistance. And what we do is we try to identify genes that have been knocked out in other plant species and try to find the orthologous genes in tomato. And we're going to talk today about BS5 and DMR6. We also have a robust collection of Xanthomonas isolates. You know, it's quite funny, actually. Maybe 10 or 11 years ago, I think the first Xanthomonas sequence was actually done. This is a bacterium, right? It's 6 megabases. It cost $1 million. Today, it costs you $5 to basically sequence an entire genome, and you can multiplex 80 at a time. So it's really changed things. So you can actually go to the field, sequence isolates, and you can actually see what resistance genes you actually need to match the corresponding effector genes that actually wouldn't be in those field isolates. So by monitoring the pathogen populations, and a lot of people are doing this for a lot of different types of diseases right now. So it's an important component. Not only do you have to understand the resistance, but you have to understand the pathogen populations and how they're changing over time. So combination of gene edits and multiple R genes. And we're not going to talk about this, but one of the dreams, 2 years ago, we published a paper where we actually solved the cryo-EM structure of a particular NLR protein bound to its particular effector called ROC1 and ZOPQ. And the idea is that we know that leucine-rich repeats are involved in specificity by recognizing pathogen molecules. And so a lot of people are very interested in making synthetic resistance genes. Can we actually start to change specificities and have it recognize any proteins? I'm not going to talk about that, but there's a lot of work around the world in many places that are trying to do that. So identify previously characterized mutants in other plants that provide bacterial resistance. So the first example would be DMR6. Identify the orthologs in tomato and create CRISPR-induced mutations. Oops. So you just basically blast some genes and do some phylogenetic analysis. It turns out there are 2 copies of DMR6. Do we go back one maybe? Let's introduce DMR6. So DMR6 was originally identified by Guido van den Aken in Utrecht, the Netherlands, okay? Where he did a forward genetic screen looking for plants that were resistant to downy mildew. And they identified a gene called DMR6, oddly, called downy mildew resistance number 6, okay? First identified, they're upregulated by pathogen infection. Inactivation leads to broad spectrum resistance. DMR6 mutants accumulate more salicylate than wild types after infection. And at least in greenhouse trials, there were no detrimental effects. So we identified 2 genes in tomato. This was work done by Daniela Tomazella in my laboratory. And again, using the tomato transformation facility with MJ and Elaine Zhang, we were able to actually make mutants. We also knew that the expression of these is also upregulated. I'll tell you what that gene encodes for in a moment. So simply, and I would reiterate what Jeff said, is if you're gonna, your edits are only good as your sequence that you have targets for. So we often will have to go in and just sequence the region that we're trying to edit to make sure that those are intact. So I'm not going to go in about CRISPR, but basically we're able to make mutations with various guides, and you get truncated proteins. Essentially, you get double-strand breaks, and then essentially you get premature termination, and you get these mutant proteins. So obviously, it was successful. Inactivation of tomato leads to broad-spectrum disease resistance to not only Xanthomonas, but Pseudomonas. It also confers a resistance to Phytophthora, which is an important pathogen, but also powdery mildews in tomato. So to make a long story short, what is DMR6? DMR6 encodes a protein called salicylate hydroxylase. So plants, when they're infected by a pathogen, essentially induce salicylic acid. But if you produce too much salicylic acid in a plant, it's going to be detrimental. So plants have evolved a mechanism to basically inactivate salicylic acid, and that's what DMR6 does. So when you make a mutation in DMR6, you don't lower the salicylate levels, and so you get increased expression of DMR6, and you get high levels of salicylic acid in these particular plants. And so that's the mechanism of how it works. We were able to quantify this. Now, when we started doing growth and development, we would see that there's a little bit possibly in the greenhouse. In the greenhouse, we actually see a slight effect on the stature of the plants. I also told you there was a gene, DMR6-2, but it turns out that gene has nothing to do with disease resistance in this case. So we didn't know that at the time. So there were two essentially orthologs. So it was DMR6-1 that was important. And this was published last year in PNAS, so you could always go up and look at this particular. So DMR6 doesn't have a role in immunity in vegetative tissues. And again, in that paper, we've done field trials down in the University of Florida with Sam Hutton and Jeff Jones, and we do see a slight off-type in those plants. You would expect that. So how are we going to deal with that? So I have a new postdoc, Young Bin Oh, who's in the laboratory now, and I think Nicholas Karavolios will be able to address this very nicely, is that what I think Nicholas has termed quantitative trait editing, where we essentially make promoter mutations so we can get a series of allelic variants. We call this a Lipman experiment, okay? Zach Lipman kind of pioneered this particular technology. But if you come to Nicholas's talk, you're going to see how it really works very nicely. So we're doing the same thing with DMR6 right now, with the idea that we want to create a whole series of alleles and find the sweet spot that basically increases salicylic acid but doesn't give off type in that particular. And I think many genes one can take this approach with. I think it's quite a solid approach. Now, another approach that we also did, there was a gene in peppers called BS5. And interestingly enough, most disease resistance genes, if you're a plant breeder, are usually dominant genes. Breeders like to deal with dominant genes so they can actually see it in the first generation. If you had to breed in a recessive gene, you obviously have to get a homozygous and go a couple of generations, so it takes a lot longer, even if you don't know what the gene is. But this recessive gene was called BS5, and typically it doesn't have the phenotype of cell death. So most dominant disease resistance genes, when you inoculate a pathogen, you get a hypersensitive cell death, which is thought to be the mechanism of resistance, but I can tell you it's most likely not, but we'll go into that later too. But anyway, this one, essentially what they did is it just lacks symptoms. So we thought this was a very interesting phenotype. So this is work by Arturo Ortega, who is a graduate student in my laboratory. And first of all, we had to collaborate with a group in Hungary to clone this particular gene. And I'm not going to go through this process, but to make a long story short, You would do it differently these days. This was done a while ago, but you'd have a resistant parent, a susceptible parent, and then you do, basically in those days, like map-based crawling if you want. But now you would actually just do bulk segregant sequencing, and you can immediately land on top of the particular gene if necessary in that case. To make a long story short, the gene was identified in pepper, and what is it? It's a bizarre gene. It's highly similar to, as you'll see, there's two genes within the pepper genome. We call it BS5 and BS5-like. It encodes a 92 amino acid protein. It has this repeat structure, and it has this cysteine-rich terminus. And the mutant was essentially an in-frame 2-leucine deletion in the C terminus. We have no idea what this protein does, but it definitely is. And what's the other thing is this has been used in breeding programs in Florida and other parts of the world, and it hasn't broken down for resistance yet. So we think it's a potentially durable type. So the idea is, can we now try to make this mutation in tomato so we could get a similar phenotype? So the way it looks like in pepper in this particular case, there's BS5 here, and this BS5-like, and these are highly, and then of course, since they're solanaceous plants, there's a lot of synteny between pepper and tomato, okay? And that, here's the BS5, and there's where the mutation is, for the recessive mutation. In this particular case, we have BS5-like. So, if you go to tomato now, you can see, well, there they are. The two genes are there in tomato. And so we didn't know what to do initially. So what we started doing is that we started to make mutations in one of them, both of them, various combinations of them. And same type of thing, CRISPR-Cas9, tomato. The nice part of tomato is that it's relatively straightforward to transform and regenerate, okay? It's one of the easier crops. It's not like cacao, I can tell you. And so we were able to get these mutations. The other nice thing is that eventually what you're going to want to be able to do if you're going to have some event that's not regulated as a GMO, as you're probably all aware, is that you use agrobacterium to introduce the Cas9 and the guide. You find your mutants, and then you segregate the Cas9 out, and you just end up with the alleles that you've created by the editing. And according to the APHIS guidelines, these essentially would be deregulated as a, not regulated as a GMO. So we made various lines into both BS5 and BS5-like, again, truncated proteins, essentially. And to our delight, we found that there was a very strong phenotype for disease resistance to these Xanthomonas isolates. And you can see it right here. This is the BS5, where the Cas9 basically has been segregated away. And we have done one field trial, and we're doing an extensive field trial this summer with Sam Hutton down in the BOM University of Florida Research Station, kind of south of Tampa. And you can do all the editing you want in the laboratory, the greenhouse, but you really have to put things out in the field and see how they perform. So we're really excited to see how these particular plants will perform in the field. So DMR6, BS5 in this particular case. Okay, the last part is work that a graduate student, Snegna Podar, did in my laboratory, and we're very interested in using ribonucleic proteins as editing reagents. And this is really important because obviously transformation by agro or bombardment leads to DNA integration. But as Jeff showed you, you can do some tricks to get away from that. Transgenes can be segregated, but it's time consuming, sometimes can be complicated. But this is really where we're interested in, such as a crop like cacao. So cacao is a vegetatively propagated plant. We're also interested in, I didn't mention, we've also developing banana transformation. And we have some very promising results with baby boom and wuschel in this particular case here for banana. So that's another monocot, but no one, I don't think, has tried baby boom and wuschel and banana yet. And if you all know that banana is being potentially wiped out by fusarium wilt. So we're very interested in that particular crop. So optimization expression of Cas9 and the guide RNA is necessary, and direct delivery of pre-complex Cas9 proteins can bypass these complexities. And of course, if you end up with an RNP edit, it's going to be DNA free. So obviously a company like Mars doesn't want a GMO, I can tell you. But if you can give them a DNA-free edit, they would be very happy in this particular case. So Snigna worked in wheat, okay? And what she developed was a very nice system. And I'll show you some of what we found some surprising results by using RNPs. So first of all, you have to decide on a target. And I'll show you the targets here are going to be 2 disease susceptibility genes in wheat. So it turns out there are some important fungal pathogens in wheat that produce host-specific toxins. And what happens is there are susceptibility genes in wheat that act as receptors for those toxins that facilitate the movement in and cause the phenotype of disease in those plants. So the idea is if you knock out those particular susceptibility genes in wheat, they become resistant. And I'll show you some results how that is. But in order to actually get this whole system running, Snigna worked out a very robust wheat protoplast isolation protocol. You can obviously get these from little seedlings. You can isolate the protoplast. And the nice thing with protoplast is that you can basically put your RNPs together and use polyethylene glycol to deliver those RNPs in. And then you can wait several days, and then you can do amplicon sequencing and really quantitate the editing efficiency of those RNPs for those particular targets that you're interested in. So Cas9 RNPs have been utilized in protoplasts of many species. This is kind of stuff that's been published in the literature. Some plants can be regenerated into edited plants. I don't think that's the case with wheat, but things like citrus, lettuce, I've also, the company Cebus, I know, can regenerate cassava protoplasts, and there are brassica components that you can regenerate whole plants back also. We also took advantage of several publications that have shown that the editing activity of Cas9 is improved with heat treatment. So it makes sense that this enzyme comes out of a bacterium that grows optimally at 37 degrees. So the higher the temperature you can do the edits, you get an increased editing efficiency at the higher temperature, and this is the case with Cas9. So this has been shown by several publications here. So systematic transfection and heat treatment of Cas9 RNPs into wheat protoplasts. So we grow the wheat seedlings, we isolate protoplasts, we assemble the RNPs in vitro, we PEG transfect, we do protoplasts at 25 or 30 for 16 hours, and we collect the protoplast pool for genotyping and deep sequencing with these particular projects. And I'll just give you the punchline. What we've been able to show that many of you that can use all kinds of algorithms to actually design guides, but you really have to test them out in vivo to see how efficient those guides will be. So we found that even there are algorithms that predict high editing efficiency, sometimes they're not so high. So I think you really have to experimentally validate the editing efficiencies of the guides that you design. And protoplast is a very convenient system to be able to do that in. Also, you have to test the viability of protoplasts drastically with temperature. So if you did these experiments at 37, you're not going to have any protoplasts left, okay? So we found that around 30 degrees was pretty optimal for survivability of the plant tissue and editing. We targeted mainly TSN1, which is a toxin sensitivity gene, and also SNN5, another one. These are kinases. This is collaboration with Justin Ferris and Tim Friesen at North Dakota State University, the ones that have cloned these genes, and they provide us purified toxins. So you can phenotype the plant, so you can just take this peptide toxin and you can infiltrate it into the leaves of the wheat plant. And if they have intact receptor, the plants will become necrotic. So it's a very rapid assay that you can ascertain. So if you knock out that particular gene, you lose sensitivity to the toxin. So this is a very convenient phenotype. So heat treatment of wheat protoplasts increases RNP genome efficiency across targets. This now has, this is in bioarchives, but it's been submitted to Plant Biotech Journal, so it's in review right now, so we'll see how that comes out. But what we're able to show that we get a much higher editing efficiency at 30 as opposed to 25. So I think if you're interested in using RNPs, you should do it at a higher temperature. But also I would check, because we find that depending on the guide, some have very high editing efficiency and other guides don't. Okay? Two publications have described DNA-free editing in meat and maize and wheat. Here, one obviously from Corteva, another one from Techargao, and they regenerated plants that had about 2.4 to 9, and either a low amount of editing, but you can regenerate plants that actually have edits. So we're interested in increasing the efficiency of editing and then regenerating plants back to be able to get DNA-free edits. Okay. So how do we introduce the RNPs? So in plants, we use gold nanoparticles, about 0.6 nanometer particles. You have to coat them with the RNPs. So you assemble the Cas9 RNPs in vitro. You absorb the Cas9 RNPs to these gold nanoparticles. We also use Transit 2020, which facilitates the uptake of those particular ones. I think it's also important to use particular RNPs that have multiple nuclear localization sequences in those particular proteins. You get a much higher editing efficiency using that. You apply gold particles to 2 bombardment discs, and you bombard 30 immature embryos. So in wheat, the way we do these experiments, we grow the wheat plants in growth chambers, we isolate immature embryos, and then we use particle bombardment of the RNPs. We treat the embryos at either 26 or 30 and 37 for 16 hours. We proliferate immature embryos on callus, induction media 28, and then we can select random 10 embryos pooled for genotyping and deep sequencing. So before you even regenerate, you can get an idea what kind of edits you're actually getting in these particular plants. Then we regenerate plants here. Oops. Can someone move it, or doesn't want to go to the next one?
The next slide? Should be.
So obviously heat treatment of wheat immature embryos increases RNP genome editing efficiency across these various targets. Like that. Now what's interesting is that when you deliver RNPs, this is work a lot that gets done on the biomedical side at IGI. If you put Cas9 into mammalian cells, the Cas9 gets degraded very rapidly. What I'm going to show you here is that in plant cells, for whatever reason, it hangs around for at least 2 weeks and continues to edit, which is quite remarkable. We think that has something to do with basically the RNP's interaction maybe with cysteine residues on the gold nanoparticles, and maybe it's coming off at a slower time, at a slower rate. So Cas9 mediated editing is sustained for at least 14 days post-bombardment. So we continue to get edits, and we know because we get new alleles arising out of these all the time. And the Cas9 can be detected by Western blot up to 14 days post-bombardment. We were quite amazed at that experiment. And then Smigna wanted to basically do experiments in protoplasts reflect experiments in regenerable tissue. So the question is, if you find high rates of editing, can you find now high rates of regenerated plants that have those edits? And is that a good proxy? So the guide efficacy in protoplasts correlates very well linearly with the efficacy in bombarded embryos. Oops. And of course here, we were able to actually, in the M0 generation bombardment, we were able to get homozygous or biallelic knockouts of these particular traits to make them toxin insensitive. So here we now have a way to actually introduce RNPs into wheat plants and to make disease-resistant plants. We're testing the phenotype. These would be toxin insensitive plants, and we assume that they will be disease-resistant in this particular case. So conclusions from this slide of the talk, and I'll end here, is that heat treatment improves RNP-mediated editing efficiency. Editing rates can be guide RNA target site dependent. When delivered by gold particles, editing is sustained for at least 14 days post bombardment. Editing rates in protoplasts have a positive correlation with editing rates in immature embryos. And protoplast RNP assays can be used as a proxy to predict stable editing rates and select optimal guides. And with 30 and 37, we get 15 to 37% plant editing rates were achieved with no selection on this particular line. So we basically add that, and we just regenerate, and then we score those plants by sequencing, and we're able to get homozygous or biallelic knockouts. So I think it's quite a good program, and I think we're currently starting these experiments in cacao right now to knock out genes. So we have the transformation, we have the embryogenesis that MJ has got going in cacao, so we're quite promising. So just Nick, who will be talking later on today, Snigna, who did the work I just told you, Donnie did the work on DMR6, Arturo BS5, Doug Dahlbeck, of course, Youngjae and his transformation team, namely Jacqueline and Elaine, and of course in North Dakota State, Catherine Running, Justice Ferris, and Tim Friesen. So I'll stop there, and I don't know if it's time for questions or we need to get going. It's up to the organizers here. Okay.
Okay, thanks. Hi, Brian. I have a question about your work with the DMR6, because you showed that even though it has a high increase in SA levels, the effect on growth is rather minimal. And we know from lots of other experiments where that is not the case, and you have high SA and it severely limits growth. So I was wondering if you could provide us with some ideas about why this might be. What is different about
there's
DMR6?
Definitely an effect, but I think what it didn't do in that paper, the PNAS paper, it didn't affect marketable yield of the tomatoes. So I don't really have a good explanation, but those plants definitely, maybe under field conditions, there are other components that come into play. We did the salicylate levels in greenhouse experiments. We didn't actually measure salicylate levels from field-grown tomatoes, so I don't know what the answer is, but I think we need to fine-tune DMR6. I think that will be a better way to proceed with that.
Hi, Brian. Nice talk. Quick questions about your comments on the Cas9 RMP actually lasts for 2 weeks. From your perspective, is that a good thing or bad thing? Because in terms of a Cas9 RMP half-lives in the weight actually lasts longer, right? Because
I think what it does, it allows you maybe in the first generation to be able to get regenerated plants without selection. And so I think it's a good thing. Now, is it a bad thing that you're going to get off-target effects? You know, we don't know. We never sequenced the entire genome, so that's maybe one downside to it. But those are all testable things that we can actually do in the future. But it's an interesting observation for sure.
For sure. Follow-up questions there. So in terms of your comments of the guide RNA efficacy, it's really correlated from your ProPast and your immature embryos. And this is just a comment. And do you think this is more of the RNP part of the effect? And if you tested the DNA vector, and do you see the same? And another thing is, in terms of efficacy, are you talking about just the efficiency, or are you talking about the allele, basically the edited alleles? They are also correlated.
I think the edited alleles, the particular guides with the targets correlate well in protoplasts and plants. We haven't done the experiment that you suggested, where we just put a plasmid in and see if it would work that way. So at least with RMPs, this is what works.
Thank you.
You have a couple of Zoom questions?
Zoom questions. There we go. How many people are on Zoom here?
Quite a few, actually. Yeah. So the first question from Zoom for you is, did you observe any pleiotropic effects arising from gene editing DMR6?
I think we were just talking about that. I think there's definitely some stature of the plants are a bit smaller in the field. So there is some effect. That's why we want to do promoter editing for that particular DMR6 and to try to find a sweet spot for that particular expression.
And then a follow-up to that was, is the heat stress given only at protoplast transfection stage, or is it happening until the regeneration stage to improve efficiency?
I think it's just done at a certain, I have to go back and look at that. I can't, whatever the slide said, I can't remember what it was. I didn't do these experiments, but I think it was just a particular heat pulse for like a day or something like that. But, and again, we can go look up, and it's in the bioarchives paper, you can check it out.
One more question, I think. Does anyone in the audience?
Jeff?
Back to the Cas9 sticking around for 2 weeks. You said you think it's due to the bombardment in the particle. Is that because you've done other experiments?
We really don't know. I mean, basically, we think that potentially, the RNP is stuck to the gold, and maybe it's just slipping off at a particular rate. Some of the bioengineers that we talked to at Berkeley think possibly some cysteine residues interacting with the gold might be important here. We don't really know the answer. But
have you looked at it like an PEG transformation and assessed if it's around in the protoplast?
I don't think we looked in the protoplast to see. We were just shocked that we kept getting new alleles over time, and we figured, well, we better look to see if the protein is still there.
Right, absolutely.
I don't know, have you guys found something similar in May from here?
I don't think we've actually looked at that.
Yeah.
We have looked at off targets with integrated plasmids, and we find very, very few off targets if you pick the guides carefully. Right. So even when you actually integrate and express kind of throughout that entire period, if you pick them very carefully, we see virtually.
Thank you very much. All right, so we will have a very short 10-minute break before we reconvene to hear a wonderful talk from Dr. Jennifer Brokey. 10:40.
Okay, hello everyone. Okay, hi. So we are ready to start our second session of the PGS Symposium. And so I would like to introduce our first speaker for this session, Dr. Jennifer Brokey, who's an assistant professor at Stanford. She received her BS in engineering from here, UC Berkeley, and her PhD from MIT, where she worked with Christopher Voigt and Alan Grossman to develop a tool for engineering undomesticated gram-positive bacteria. For her postdoctoral research, she was at Stanford with our Carnegie Science with Jose Dineni, engineering spatial patterns of gene expression across plant tissues using synthetic genetic circuits. And now she's interested in applying synthetic biology to address climate change issues and enhance sustainability. So Dr. Brokey, I'll give you the floor. Thank you so much for coming.
Awesome. Thank you for the introduction. Awesome. Hi, everybody. So I'm going to talk primarily today about work that we just published. This is work from my postdoc where we were engineering synthetic genetic circuits to modify plant development. So I'm interested in engineering the size and shape of plants, because in plants, like in all biological systems, size and shape are important determinants of function. And if we can figure out how to engineer these features, I think we can create some new and really useful plant varieties. On this slide, I'm showing two examples in which humans have already changed the shapes of plants to produce something useful. On the top is modern maize, which was bred from its wild ancestor teosinte to increase both the size and the number of seeds that the plant produces. And these changes in maize have made it into one of the most productive food crops we have today. On the bottom is a dwarf rice plant, which helped usher in the Green Revolution by allowing farmers to apply massive amounts of fertilizer to their field and get an increase in grain yield instead of just more straw biomass. Now, what's exciting about these two examples is that their changes in structure are both the result of changes in DNA sequence. And as a genetic engineer, I see this link between sequence and structure as an opportunity to apply some advanced genetic engineering techniques towards changing biological shape. It's really echoey. Okay, so I'm not just interested in engineering shape in order to create a more productive maize variety, although that would be really exciting. I'm actually interested in engineering structure because of how important it is for environmental stress tolerance. The shape and angle of a plant's leaves can affect photosynthetic efficiency and the ability of a plant to tolerate heat stress. The shape of its root system determines its ability to acquire water and other essential nutrients from the soil. And structures like the air channels that you can see in these wetland plant stems are essential for survival in flooded soils. So if we can engineer structure, we might be able to engineer crops that are more resistant to the environmental stresses that we know are coming in the face of climate change. But if the goal is to engineer structure, the question becomes, how do you do it? Well, what we know is that engineering structure is essentially engineering development, and that engineering development requires very precise spatial control over gene expression. To illustrate this point, I'm going to show you 3 different Arabidopsis plants that are growing on agar plates so that you can see the shape of their root systems. This first Arabidopsis plant is the Columbia ecotype. It's essentially wild type. It has a long primary root that grows down in the direction of gravity. It has several lateral root branches that also grow down in the direction of gravity. And if you were to zoom in on this root system, you'd see that it's covered in these tiny root hairs, which are structures that are really important for water and nutrient acquisition. Now, if you make a single base pair change to this plant that modifies the amino acid sequence of a developmental regulator called IAA14, you make several different changes to the plant structure simultaneously. This mutant plant has no lateral roots. Its primary root is a little bit shorter, and it has a hard time sensing gravity, which you can tell because the root does not grow straight down. It's also missing root hairs, and there are several structural changes to the shoot. Now, one of the reasons why this mutant transcription factor, this mutant developmental regulator, can affect so many plant structures simultaneously is because it's expressed in all of these different tissues throughout the plant. But if we instead express this same mutant regulator from a promoter that's only active in one cell type, we can make a really targeted change to plant structure. So here, the plant does not produce any lateral roots, but it's otherwise identical to the wild type plant on the left. So our strategy is to reprogram structure, and in this project, root structure, using very precise spatial control over gene expression. Now, if you want to have precise spatial control over gene expression, there are a couple of options. You can look for promoters that have expression patterns that are ideal for the type of engineering you want to do by searching through the literature. You're lucky if you work in Arabidopsis groups like I do, because there are lots of promoters whose expression patterns have been pretty well characterized. So you might be able to find something just by looking in the literature that has an expression pattern that you want. But if you can't find something in the literature that has an expression pattern that might be useful to you, you might try mining an RNA-seq data set in order to identify genes that have an expression pattern that you think would be useful, and then take the promoters from those genes in order to express your genes of interest. Now, that seems simple on its face, but in practice, it's actually somewhat complicated. And I like this paper because it shows how this can be complicated. These authors tried to take the promoter for ARF7, which is a gene that they know is expressed in the root tip of Arabidopsis, and use it to express a fluorescent protein. When they took the 5' genomic sequence for this gene and fused it to a fluorescent protein, they saw no expression in the root tip of Arabidopsis. But when they instead went back and took not just the 5' genomic region, but the first intron and exon as well, and fused that whole sequence to a fluorescent protein, they recovered the expression in the root tip of Arabidopsis that they were expecting. Now, as an engineer, this is somewhat problematic because that means that I have no idea what region of the genome to use as a promoter when I want to express a new gene in that same pattern as the original, right? It might be that I could take the 5' genomic region, but it might be that I would need sequence from elsewhere in order to recover an expression pattern. Okay, so if there's no published promoters in the literature that have expression patterns that you want, and it might be challenging to mine promoters from RNA-seq data sets, what can you do? Well, what I thought is that we can use logic gates to generate new spatial patterns of gene expression. I'll illustrate to you how this would work using cartoons and fake promoters. Here you're looking at cartoons of the root tip of Arabidopsis. There's 2 promoters shown here. Promoter A has an expression of a fake fluorescent protein in the root cap of Arabidopsis. Promoter B shows expression in a subset of root cap cells called the columella and in some central vasculature cells. So if these are 2 promoters that you found in the literature, you combined their expression using a logical AND gate, you should get expression only where promoter A and promoter B are active or in those columella cells. So you're able to generate a new pattern of expression using 2 published promoters and a logic gate. Now, there's not just one 2-input logic gate, there's actually 16 different 2-input logic gates, and you can build on complexity by incorporating even more tissue-specific promoters. So in theory, if you can build logic gates in plants, you can start to build lots of new spatial patterns of gene expression. Now, in order to build logic gates in plants, we first started by building some tools to control gene expression. These tools that we've started with are synthetic transcriptional activators and repressors. They're really simple. The activators are composed of DNA binding proteins fused to activation domains and nuclear localization signals. These transcription factors, when bound to the DNA, should recruit RNA polymerase and initiate transcription of downstream genes. The repressors, in contrast, are just DNA binding proteins fused to nuclear localization signals. When these are targeted towards regions of the genome that would otherwise act as a promoter, they should prevent the assembly of RNA polymerase and prohibit the transcription of downstream genes. So we built libraries of synthetic transcriptional activators and repressor proteins, and then tested to see whether or not they were functional using transient expression assays in Nicotiana. I'm sure a lot of people in this room are familiar with transient expression assays, but when I started in plants, I thought that this was a shocking type of experiment, because what you do is you take a plasmid, you transform it into agrobacterium, and then you infiltrate them into plant leaves, right? Which means you fill the airspace of the leaf with this solution of bacteria, and they do this trans-kingdom conjugation of DNA from the bacteria into the plant cell, and then the plant cell expresses those genes. That's wild. I think that's cool that that works. So we use this type of assay to see whether or not our transcriptional activators work by infiltrating 2 different types of constructs into Nicotiana leaves. On one side of the leaf, we do just the synthetic promoters driving expression of GFP, and on the other side, we infiltrate both the synthetic promoter driving GFP and the synthetic activator. What we find in most cases is that we get strong expression of GFP only when the activator is infiltrated into the leaf along with the synthetic promoter, which means that activator is able to turn on the synthetic promoter. We have a constitutively expressed M-cherry on the plasmids that we infiltrate, and this allows us to normalize these GFP measurements to M-cherry in order to control for any technical issues that might arise during these infiltration experiments. We quantify, oh, there we go. Okay, so we quantify fluorescent protein expression by taking leaf punches and placing these leaf punches in 96-well plates and then measuring fluorescence in a plate reader. And for the rest of this talk, I'm going to actually be reporting this ratio of GFP to M-cherry. Okay, so we built out 10 different synthetic activators and promoters that the synthetic activators should be able to turn on by oligomerizing the binding sites for these synthetic activators. And then we tested all combinations of these in Nicotiana leaves in order to see whether or not our synthetic transcriptional activators were able to turn on gene expression at the promoters we designed them to turn on, and whether or not they were specific to those promoters. We found a subset of 7 that were both active and specific, and the specificity is helpful for us to build out logic gates where multiple synthetic transcription factors are trying to turn on specific promoters all within the same cell. In some cases, the synthetic activators weren't as strong as we would have liked them to be, which means they didn't turn on as much GFP expression as some of the others in the set. And so we wanted to try a little bit of simple protein engineering to see if we could get these synthetic activators to be more active in plants. We swapped out the activation domain that was originally fused to the bacterial DNA binding protein in order to improve them. The activation domain that we started with was BP16, which is sourced from human herpes virus. This is an activation domain that had been shown to function in plants before. It's kind of amazing that it works as an activation domain across so many different types of organisms. And when we swapped it out for an activation domain from a plant transcription factor instead, we saw an increase in how active the synthetic transcription factor was. So we think that we could do a little bit of simple protein engineering to start to tune gene expression levels. We tried a similar type of thing with the repressors because the first version of these repressors that we built were actually not very good. On this slide, what you see are the synthetic promoters that we built to be repressed by our synthetic transcription factors. These promoters were full-length 35S promoters with a single binding site at the 3' end. And when we tested to see whether or not these promoters could be repressed by transcription factors, we saw that the vast majority of them were not repressible. We thought, okay, maybe it's the mechanism of repression that we were trying to introduce here, that steric hindrance wouldn't work well if the protein was targeted to the 3' end of the promoter. But maybe if we add repressive domains to these transcription factors, we could get better repression. And so we tried fusing some well-known repressive domains to these transcription factors, but saw that that had a pretty limited effect on our ability to turn off the 35S promoter. What did work well was moving the binding site for these transcription factors to somewhere else in the promoter. In this case, we tried targeting regions that were closer to core promoter elements within the 35S and found that moving a single binding site internal to the 35S promoter gave us a 7-fold repressible promoter, and then adding a second or third repressor binding site gave us even more repression than that. Okay, so this is just showing that we can do a little bit of tuning of gene expression and that we have some tools for controlling gene expression in plants that are functional. The question becomes, how do you use them to build logic gates? So I want to walk you through the design of a biological AND gate so you have an idea of how these things can be put together. In this case, we are using GFP as our output for the logic gate, and we should only see GFP expression when our 2 inputs are present in the plant cell. So in our case where we have no inputs, there's a constitutively expressed repressor targeted to GFP's promoter that's keeping expression of GFP off. Our first input is a synthetic activator that would turn on expression of GFP, except that that repressor is there keeping expression of GFP off. Our second input is a repressor that turns off expression of our first repressor. But because there's no activator present in the system, there's no GFP expression, and it's only when you have both input A, the activator, and input B, the repressor, delivered to the cell that you should see expression of GFP. We test to see whether or not this type of design works, again using transient expression assays in Nicotiana leaves, except in this case we infiltrate 4 different combinations of strains into the leaf. In the first spot, we have just the output plasmid delivered, so this is the no input case. In the second spot, we have a plasmid that encodes for the output and for a input transcription factor. In the third case, the second input, and then in the end, you have both input transcription factors being delivered. What we see is that you get strong GFP expression only when both of your input transcription factors are delivered to the plant cell. We can quantify gene expression, again, using plate reader assays. But what I think is exciting about the tools that we've developed is that you can build not just AND gates, but actually a bunch of diverse logic gates. So this is showing you designs for 15 different 2-input logic gates and how they might be constructed using the tools that we've already built. To date, we've been able to build 14 of those 2-input logic gates and show that they function well in Nicotiana leaves. Now, building logic gates in leaves is not getting us that much closer to being able to control spatial patterns of gene expression in roots. So what we've done is instead of just constitutively expressing the input transcription factors, we've turned to using tissue-specific promoters to control their expression. So now these 2 tissue-specific promoters are serving as the inputs to our logic gates. The 2 promoters that we've used so far are the sombrero promoter, which drives expression of GFP in the root cap of Arabidopsis, and this pin-4 promoter, which is active in the columella and some central vasculature cells, actually exactly like the cartoons I showed you earlier. And in theory, when you combine the activity of these 2 promoters, you should be able to get several different patterns of gene expression. I'm showing you here cartoons for, I think it's 7 or 8 different logic gates in Arabidopsis, and then the resulting patterns that we get when we deliver or when we try to implement these circuits in Arabidopsis. we're starting to be able to control gene expression patterns in Arabidopsis. I want to zoom in on a couple of these gates to tell you a little bit about them. So the first one I want to focus on is this buffer gate. So buffer gates are extremely simple. They should just recapitulate the expression pattern of the input promoter. So the design for this buffer gate is shown along the bottom. We basically have the tissue-specific promoter driving expression of an activator that in turn drives expression of a reporter gene. When we use sombrero as our input promoter, we can really nicely recover the sombrero expression pattern using a buffer gate. Now, why would you want to do this? That's a seemingly trivial thing to do. But what we found is that we can actually control expression levels by changing the number of binding sites in the output promoter. So relative to just directly driving expression of a fluorescent protein, directly driving expression of a fluorescent protein using a constitutive promoter, if we have that same promoter driving expression of an activator and then a synthetic promoter with multiple binding sites for that activator, we can amplify the amount of GFP that we express. Or if we have just one binding site, we can actually repress the amount of GFP that we express relative to just the input. So this could be a method of tuning gene expression in plants, right? And when I thought back to these plants that I showed you at the beginning that have no lateral roots, I thought, well, wouldn't it be nice if we could tune the number of lateral roots that these plants produce? An Arabidopsis that has no lateral roots does pretty poorly. If you plant them in soil and you damage the primary root, they usually die. If you tug on them, they just come flying out of the soil. And really, having lateral roots is important for anchoring them into the soil and for helping the plants obtain nutrients and water. So wouldn't it be nice if we could control the number of lateral roots that they produced? I thought, well, maybe we can apply this gradient in promoters in order to develop a gradient in root branching. You know, we know if we express a lot of this developmental regulator, we can completely repress root branching. But if we tune it down, could we get a gradient in root branching? Should say that the other thing that we might identify is just that there's a threshold, right, above which we are repressing root branch development and below which expression of this developmental regulator will have essentially no effect. We wanted to build these systems to find out. So the overall structure of this looks like this, where we have the tissue-specific promoter driving expression of the activator and then this gradient of synthetic promoters driving expression of the developmental regulator. When we first implemented this in Arabidopsis, we saw that none of the plants produced any lateral roots. It didn't matter how many binding sites we had, we were producing presumably too much of it in order to see any different phenotypes. The really nice thing about using bacterial DNA binding proteins as the basis for these synthetic transcription factors is that we have a really good idea of what binding sites they prefer. So this is data collected from Corynebacterium, which is where this bacterial DNA binding protein is sourced from, showing which bases are really important within its binding site for that protein to attach to DNA. Knowing this allowed us to make a really targeted library of mutant binding sites that had less and less capacity to recruit the synthetic activator. And when we tested them in Nicotiana leaves, we saw less and less GFP expression with the mutant binding sites. So we can make promoters that were even less active. And when we applied bees in that same system, we were able to generate this gradient in root branch density. So this has less and less expression, we think, of the synthetic activator as we develop more lateral roots. So we can quantify root branching, and then we actually went back and also quantified some other aspects of these root systems, like how long the primary root was, whether or not they had root hairs, and their gravitropism, how well they sense gravity, to show that the mutations that we made or the changes that we made were specific to root branching. So we're excited about this approach for being able to control structural aspects of plants. But that was just one of the simple logic gates, right? Why build out even more complicated logic gates? And I want to answer that by talking about not implies gates, because I think not implies gates are really cool. This type of gate is on only when one of your inputs is present in the system. As soon as your second input is introduced, the gate shuts back off. What that allows you to do is take one promoter's expression pattern and subtract out another, right? If the gate goes off when the second input comes on, you can get an expression pattern that is refined relative to either input promoter. What we would like to do is try to use this particular expression pattern generated by the Symbreo and PIN4 promoters that we had to control gravity sensing. So in Arabidopsis roots, the model for gravity sensing is that you have these starch-filled granules in the columella that are heavier than the cytoplasm. And when the root is reoriented, these granules fall and tug on actin filaments within the cell. Tugging on these filaments releases auxin asymmetrically into the neighboring tissues or into the neighboring cells. And the high amount of auxin that ends up on the underside of the root prevents those cells from elongating, whereas the small amount of auxin along the top side of the root system has no effect. And that asymmetry in growth allows the root to bend and reorient itself with respect to gravity. And we thought, well, we have this mutant developmental regulator that represses auxin signaling. We didn't go into it, but that OXIA, that IA14 represses auxin signaling. And we thought, well, if we can express it in these neighboring cell types, maybe we can change the plant's ability to respond to gravity and alter its relationship with gravity. We're still early on in figuring out whether or not we can do this, but I wanted to show you some preliminary results here. So along the top on this slide are wild type Arabidopsis. These little seedlings, sense gravity, their root systems go down in the direction of gravity. If we express the mutant IAA from the Symbreo promoter, we can get plantlets that can't sense gravity well. This one's actually growing directly up. But if you were to zoom in on the root cap, you would see that its development is pretty messed up, right? The tip develops as this big ball of cells, and on the far right is a reporter for auxin signaling. You can just see how kind of messed up the development of the root tip is. We know that correct auxin signaling in the columella is important for for the root tip to develop correctly. So when we repress expression of that IAA14 just in the columella region, we recover a more normal looking root cap where the plant is not able to sense gravity well. These also still grow agravitropically. But the root tip, although more like wild type, isn't completely normal. And so we're starting to figure out whether or not there's incomplete repression of the IAA14 gene in the columella region that is giving us this sort of still slightly abnormal root cap, or if it's something else entirely. But we're excited about trying to use these tools to better understand how plant development is working and for being able to make targeted changes in the plant structure. Okay. Everything I've shown you so far is in Arabidopsis and Nicotiana, but we would love to move these tools into other plant varieties. I'm showing here sorghum, but we're interested in grasses in general. But we know that control over gene expression in grasses is different than control over gene expression in eudicots or even just in Arabidopsis. And so how do we make this leap? How do we go from tools that we have that are functional in Arabidopsis to other plant species? One of the things that really helped us do this engineering in Arabidopsis was having this transient expression system in Nicotiana where we could test all of our components and get a good idea of what might be functional when we move into Arabidopsis. And so in order to move into other plant species, we're turning to protoplasts. And we're starting now with protoplasts of Soteria to test to see whether or not our synthetic transcription factors are going to be functional in Soteria cells and whether or not the same type of auxin mutant can be made in order to modify development or repress auxin signaling at least. So this is really preliminary. This is technical replicates from one experiment where we are testing a single bacterial DNA binding protein fused to different activation domains in Soteria protoplasts and finding that the type of activation domain that we fused to them seems to matter in terms of how strongly these synthetic transcription factors can turn on the synthetic promoter in grass cells. But this is just the beginning of us starting to test some of these tools in grasses, and I'm sure the story will change as things progress. But we're excited about engineering other plant species in the future. I am excited about engineering bacteria, but we haven't worked on anything like that in my lab yet. And once we've made engineered plant varieties, we would like to test how the changes in structure affect the plant's ability to survive in challenging environments, so somehow closing the loop on some of this. Okay, so I want to end by thanking Jose and the rest of the Genome lab, where I was a postdoc and did most of the work I talked about today, and then some of the people who are helping me build out my lab. Not all of those people are in my lab, but we are in a very supportive environment at Stanford, so they hang out with us and we buy food. And thank you guys for your attention.
So we have time for a couple of questions, if that's okay?
Sure.
Do we have any questions from the audience?
Thanks, and thanks for an amazing talk. We were wondering when we read this paper about the choice of pin and sombrero. What made you guys choose those? And if you were to choose it again, would you choose the same ones or different ones?
Yeah, I chose them actually. The choice for them was motivated by that gravity sensing idea. I really wanted to be able to control the number of root branches that a plant produced, how quickly the primary root grew, and then the angle with which the lateral roots grew with respect to gravity, which was a really ambitious place to start a postdoc from. But I chose those because I had read papers about how gravity sensing worked, and I thought it would be really interesting to try to isolate that region of the root cap in order to try to modify gravity sensing. Now, would I choose those same promoters again? No, I don't think so. PIN4, it is important for auxin transport, and so trying to express an auxin-insensitive regulator using that promoter can have weird feedback effects that complicate interpreting your results. Also, the columella region itself, the starch-filled granules, can make imaging in that region more challenging. But I like sombrero. I thought sombrero was cool.
Hi, awesome talk, thank you. I'm so intrigued at how, as you stack activating motifs, you just see the continual increase in expression. Is there a numerical limit at which you can increase expression?
I'm sure there's a limit, yeah. Not just from a functional point of view, but I'm sure you hit some sort of silencing limit. We don't have a good sense for how many of those activating domains or binding sites are actually bound by anything at any one time. But we haven't probed yet, so at least in our hands, we don't know what the limit would be.
Well, thank you so much for coming today.
Excellent, great. Thank you very much for this really kind introduction. And thank you to the organizers for giving me the opportunity today to share with you some of the work I've done. Some of the work I've done as a postdoc in Chris Niogi's group in the Department of Plant and Microbial Biology here at UC Berkeley. So my discussion today will focus on diatoms, a widely distributed group of photosynthetic single-celled algae. And it is deemed that their ecological success is due to, in part, their really highly ornamented silica or glass-based cell walls. So on your right, you can see a micrograph showing an arrangement of 52 cleaned diatom cell walls. You can appreciate their diversity of their morphologies. So diatoms are actually responsible for a fifth of planetary carbon fixation, and they serve as model organisms to study biogenesis and morphogenesis of biominerals, a topic I have been fascinated by for many years now. And so just as a quick definition, a biomineral is any material produced by living systems that contains an organic and inorganic phase. So the most relatable one being bone. So the primary model marine diatom for studying these types of questions is Thalassiosauria pseudonana. It's been sequenced, and you can appreciate its morphology down here on this SEM image. So diatoms are by far not the only glass makers in nature. There are many organisms that can produce silica across the tree of life. And for today's discussion, I want to emphasize that silicon, actually in the form of silica inclusions called phytoliths, is also very important for plant health. So in this study on your upper right, they studied, investigated the morphological diversity of these phytolith inclusions in rice. So there are many reasons why one would want to study biosilicification in diatoms. Material scientists have for many, many years been fascinated by the hierarchy, porosity, and high surface area diatom shells contain, which are highly desirable features for engineering and designing applications using diatoms. Related to that, given that silica is a biocompatible material, diatom shells have actually been used and explored as a way for targetedly deliver drugs. Of course, we should not forget their crucial importance in cycling elements globally. So they are obviously important for the silicon cycle, but I also want to emphasize their crucial involvement in carbon drawdown via a process called biological carbon pump. And last but not least, we are actually standing on large deposits of a rock formation called the diatomite, which is actually made out of fossilized diatoms here in California. Okay, so in the spirit of this symposium, I also want to argue that investigating silicon metabolisms across the tree of life, including diatoms, may provide a future avenue to design novel strategies to engineer climate-resilient crops. So as I mentioned before, silicon is not an essential element for plant health, but definitely highly beneficial. So in the studies in rice over the past 2 decades, it's been shown that when a silicon uptake is disrupted, that highly negatively affects grain yield, tolerance to insect damage, pathogen infection, and tolerance to various abiotic stresses, including drought. So now that we are all sufficiently motivated to study diatom biosilicification, I want to describe to you a novel method to engineer diatom genomes using Thalassiosauria pseudonana as the model. So, but first, how do we actually introduce genes into this organism? So we actually use bacterial conjugation. In this particular experiment, we fused a known glass-associated, silica-associated protein called silafin-3 with the green fluorescent protein and successfully expressed it in this diatom species. And throughout today, I would be happy to share with you a video of a three-dimensional reconstruction of these fluorescent cells. So please find me today, and I'm happy to share that with you. Okay, so we then sought to identify a good appropriate target to develop our genome editing approach, and we actually turned to another really fascinating aspect of diatom metabolism, which is nitrogen metabolism. Specifically, we zoomed in on one enzyme encoding gene called the urease, which hydrolyzes urea in ammonium and CO2. So in a previous study, in a different model marine diatom called Phaeodactylum trochonurum, a urease knockout cell line was shown not to be able to grow when urea was present as the sole nitrogen source. So we thought that was a good gene to develop our genome editing approach. So moving forward, I now want to introduce you to the design and assembly of these bacterial conjugation compatible vectors for multiplex genome editing using tRNA, gRNA arrays in this model marine diatom. So under A, you can see a schematic of these designed and assembled vectors. that contain elements for maintenance and replication, both in bacteria and a diatom. In addition to Cas9 tagged to YFP, the most important crucial element is highlighted below, which is effectively a U6 promoter-driven cassette that contains an array of tRNA and gRNAs. I'm sure a multiplexing strategy many of you are familiar with. So effectively, in this particular style type of multiplexing, we exploit the very conserved machinery of eukaryotic cells to process pre-tRNA into tRNA. So what happens is when this particular array gets expressed, a primary transcript gets processed by endogenous RNase, a combo of RNase P and RNase Z, which liberates the single guide RNAs for multiplex editing. So we're super happy to report that this particular approach works in Thalassosauria pseudonana. In A, you can see our Cas9 cut sites mapped onto a schematic of the urease protein. The deletion, which is a 37 base pair deletion, leads to shortening of the enzyme to approximately 1/9 of its size and complete inactivation of the protein. Under B, in the top, you can see that when these knockout strains are genotyped, where we basically amplified the region around the cut sites, you can appreciate the shorter product in knockout lines relative to the wild type, and that was also confirmed using Sanger sequencing, showing a very clear deletion as expected. Now, we then wanted to move from this genotype success on the genotype level to the phenotype, and we asked how do mutant lines grow in the presence of various nitrogen sources. So we tested a nitrate as a default nitrogen source, urea, and a no nitrogen control. And turns out that, as expected, the newly zygous Thalassosauria pseudonana mutants did not grow on urea. So under A, we have a qualitative outcome, and under B, a quantitative outcome, where basically you can appreciate this is a bird's eye view of deep well plates from this growth experiment. In the presence of urea, only wild type can grow just fine, whereas all the knockouts were unable to grow on urea. Conversely, in the presence of nitrate, all the strains were growing just fine. In a no nitrogen control, none of the strains were growing as an important control. And here in B, you basically can see a quantitative outcome of this, a growth curve showing, if we focus only on the pink lines, you can appreciate again that the wild type strains were able to grow in urea versus the knockouts which were unable. So with that, basically what I share with you today is obviously just a proof of concept. It's the beginning of what I think is going to be a long journey that will lead us to a wealth of insights into the fascinating biology of this particular model diatom and obviously also other marine diatoms. To put it in slightly more concrete terms, this work represents the first time tRNA, gRNA arrays were used for CRISPR genome editing in a microalga. Second point is a little bit more, less of a conclusion, but something perhaps a food for thought for all of us is it's been shown in the in Phaeodactylum tricornutum, in the other marine diatom I mentioned, that such a knockout actually leads to a urea buildup. So could we envision ways to use these strains as a perhaps sustainable way to produce urea as a fertilizer? And of course, concurrent editing of multiple genome loci opens up many opportunities. So obviously it facilitates the study of genetic interactions involved both in silica cell wall biogenesis and morphogenesis, but also other questions we're interested in diatom field. It opens doors to combinatorial genetic perturbations as perhaps a new way to explore the available silica patterning space, and thus actually engineering new morphologies for applications in catalysis, filtration, and many other fields where diatoms are interesting. Okay, this basically brings me to this last slide where I just want to give you a necessarily superficial overview of what I envision doing both as a postdoc here at UC Berkeley and hopefully sometime soon also in my own independent research group. So there's many questions that we're interested in when it comes to polyamine metabolism. So these metabolites are a fascinating bridge between nitrogen and silicon metabolism in diatoms, where in addition to the core homeostatic functions, these metabolites are also structural elements of the final biomineral. So we actually want to focus on looking at specific set of enzymes involved in these processes that start with the non-proteogenic amino acid ornithine. We're also very interested in how silicic acid, which is the primary building block of silica biominerals, is actually sensed by diatoms and what kind of downstream signaling events are involved in this sensing. And of course, there's actually a really amazing effort ongoing as we speak, sequencing many other diatom species. So we would also want to be we're also very interested in developing new model marine diatoms for a variety of future studies. And with this, I want to thank Chris Niyogi, the whole Niyogi Lab and HHMI for their help, support, and funding, and many other individuals that were crucial for my work, both related to the project I described and other projects I didn't have time to talk about today. And of course, thank you very much for your attention. And yeah, the future of diatom biology is really bright. So I'm very excited to keep going. And with that, yeah, thank you and happy to take any questions.
Hi, thank you. I have it's kind of a technical question. When you use the tRNA approach, I'm curious, were you using rice sequence for the tRNA?
Yes.
Yes? So do you think that if you use from the, how do you say it?
Yeah, Thalassosauria. Yeah, yes. We, in fact, yeah, I didn't mention, but I basically have also built vectors having actually 2 different flavors of glycine tRNA from Thalassosauria. Cannot really at this point say whether they work or not. I'm yet to test them, but yeah, I've taken a look at tRNA sequencing sequences in Thalassosauria.
The rice one.
Yeah, but the rice one is the one that led to the results presented.
I guess this isn't maybe an overly broad question, but when you look at all the different domain, like clades that are using silica, are diatoms, like, do they stand out in any way, or are there really like that elaborate silica structures that exist in other life forms too?
I mean, there are definitely other species, even within the stromanopyle part of the tree of life, where there's organisms that make like effectively cocoliths. So cocoliths are actually calcium carbonate precipitating algae from oceanic algae, but there's a related pathway that gives rise to silica plates. So those are also species specific in this particular stromanopyle lineage. So I think I would say, yeah, diatoms definitely, with respect to species diversity, the estimate is around 100,000 species, and I think they probably, I would argue they lead the way when it comes to like ornamentation and diversity, but that is not to say that there's like other, definitely other silica producing organisms with a variety of morphologies. Yeah.
Thank you so much.
Good.
Also, do we have the laser pointer still around? I think. Is there one up here? Oh, we do. Good. Okay. Well, Yorne, thank you so much. Now I have the pleasure to introduce Dr. Ryan Nasty. He is a postdoctoral associate here at the Blackman Lab in the Plant and Microbial Biosciences Department. His overarching research goal is to combine genomic trait prediction with next-generation plant genome editing and transformation techniques. During his PhD in the Voitis Lab at the University of Minnesota, He was a part of a team focused on implementing developmental regulators for the prospect of improved plant transformation. And currently, he will tell us a bit about his work today on transforming sunflower. So Ryan, please come to the stage.
Hi, I'm Ryan. I'm going to talk today about my work in plant sunflower engineering, and I want to thank the organizers and everyone here for attending. And so I want to start by talking about our crop system, in so much that throughout human history, humans have taken wild plant species and converted them in many different facets to useful crops. And so examples of this come in the form of taking wild multi-branch sunflowers and converting them into these single-headed, much larger crop varieties. Throughout breeding history, we've converted tomatoes from these tiny berries to the much larger consumer-based food crop we see in stores. And then in the classical model, we see the transition from the hard-shelled multi-tiller teosinte into the soft-kerneled, single-stalked maize. And so this transition begets two major questions, namely, what types of traits do we want to see in our food crops, and how do we make these such food crops? And so I'm going to start by talking about the types of traits that we are selected upon during breeding of our crops. And so there's two major flavors of these types of traits: consumer traits, which includes different flavor varieties, different colors, useful properties for the human consumer in their own day-to-day lives, as well as agronomic traits, where you want to reduce the amount of seed loss, increase the total number of seeds so that you're increasing overall yield. And so looking at sunflower as an example, we have on the consumer side of traits, improving oil content so that you create a healthier sunflower oil for use. And then on the agronomic side, you would look towards increasing seed numbers so you increase overall sunflower seed yield. But this begets the question, how do we build upon this trait variation? And so I want to walk through a specific example of de novo domestication of the wild tomato relative Solanum pimpinellifolium. And so this research group took guides targeting the genes multiflora and fasciated, and were able to push for more floral branching, so you improve the total number of fruits on a given plant, as well as increasing the locule number, so you end up with a larger fruit. And so looking at this prospect holistically, you have your target plant species, in this case, Solanum pimpinellifolium, and you want to improve floral branching. You can then design your gene editing reagents and run through this vague plant generation pipeline to, on the other end up with your edited plant that has your trait of interest. Unfortunately, it's not this wide funnel. You have multiple blocks in this process that serve as key bottlenecks to generating these plant varieties, namely in the reagent delivery and plant regeneration aspects. And so where these major bottlenecks come of age is in the tissue culture based regeneration process. And so how this works is you'll take your seedlings grown in sterile culture, you'll cut them and create these explants that you can then deliver your reagents of interest to using different hormone regimens. You'll create callus structures that can be progressed to shoots and eventually into rooted plantlets. This prospect is tedious and technically challenging. This takes a long time and is not applicable to all plants or even all cultivars of all plants. And so this really brings up the main point, is we need ways and thinking of how we can improve these processes across plants. And so as alluded to earlier today, there have been many pioneers in the avenues of using developmental regulators to push towards new methods of reliable plant transformation, namely from Bill Gordon Cam with the baby molluscal somatic embryogenesis. But we built an orthogonal system wherein we built this in the Nicotiana seedlings, where we have an agrobacterium treatment that we co-culture with whole seedlings in liquid culture. that then can allow for de novo growths to form, eventually progressing to shoot-like structures and eventually rooting in whole plantlets. And so the example I'm going to walk through in practice here is with the combination of bushel and shoot stemless, where we track delivery with a luciferase reporter and look for sites of high expression of relating to our constructs of interest. We then begin to see these globular growths form, eventually beginning to differentiate with these little leaflets. And over time, we end up with full shoot-like structures forming on the cotyledon edge of these different seedlings. And so we developed this in a multiple different, two different main approaches. One where this the co-culture seedling approach I just walked through. But we additionally built a direct injection platform where you can cut full in-soil grown plants down to initial node. You can deliver your agrobacterium at that cut site, and eventually you'll create a new shoot that will form from that site. And so in practice, we were able to show successful transgenic generation in Nicotiana, as well as editing of this PDS gene that gives you this white locus. And in tomatoes, we were able to get single generation, non-transgenic, gene-edited tomatoes that present with this yellow-white or yellow phenotype related to the knockout. In addition, with the direct injection, we were able to also get these white nice shoots that were forming, but additionally, we were able to see editing and luciferase presentation in tomato shoots that we were inducing. And so now I want to switch to the platform I'm testing currently, and that's applying these developmental regulators in sunflower. And so sunflowers are one of the more challenging species to transform. And so if we can take our developmental regulators, apply them to sunflower, hopefully we can get this pipeline up and running so that we can eventually establish efficient transformation towards these traits of interest, oil content, and increased seed yield. So I want to walk through two short stories where one, we're working at applying these developmental regulators so that we can get this transformation up and running, but also shifting a bit outside of the transformation space and looking at given genes that we want to target for this modification towards those trait goals. And so first, I'll walk through my progress with applying developmental regulators for shoot regeneration. Since sunflower seeds and seedlings are much larger and have these waxy coats, we had to shift some of our techniques that worked very well in Nicotiana and tomato towards a sunflower cotyledon-based system. And so what we would do is take sterilely grown seedlings, isolate the individual cotyledons, and then perform the co-culture on that combination with the agrobacterium as opposed to whole seedlings in culture. And then we're working through the progression from those tissues towards growths and eventually shoots. And so at this point, that's too far. At this point, we've tested a variety of different developmental regulator combinations with certain aspects, certain combinations working quite well in our hands, but we're using the Ruby reporter to track growth. And as you can see here, we have differing levels of growth from effectively no growth to a more amorphous, less controlled growth, and eventually down to the highly controlled, really nice globular growths that we're hoping to see shoots progress out of. And so at this point, we have successful growth induction with the developmental regulators tracking these Ruby positive, pigment positive reporter sites, but we're working towards methods to get actual shoot progression out of these sites. Shifting now towards the direct injection application, I've been taking seedlings grown in tubs and cutting them down to their initial node sites and delivering some of these same combinations of regulators and tracking for Ruby growths as in the co-culture setting. And so what we've seen thus far is you get a majority of wild type shoots that occur first, and you'll see this initial peak of overall shooting, but over time you get to see these shoots that have either partial Ruby sections or of more recent, we've been tracking and seeing majority or full Ruby positive shooting events, which over time, after that initial wave of wild type shoots form, we see more and more of these ruby positive sectors forming. And so we're preliminarily on our way towards nice, reliable regeneration in these sunflower systems. But now shifting out of the transformation side of things and more towards key genes for domestication, when looking at traits like oil content and seed yield, where do we start for our genomic predictions and genes to modify? And in lab, we have the wealth of information that's been collected from representative wild sunflowers, land races, and even DNA from ancient archaeological sunflowers that were collected at field sites from Native Americans. And so we can take and leverage the variation in the genomes from across these different samples through both diversity and time to identify key features for through domestication for manipulation. And so from previous members in the lab, they've dwindled this full genome down to 413 major gene candidates that we're going to look at across a variety of different trait groups, including fatty acid metabolism, as I've been talking about, towards a major trait we want to look at, as well as RNA-seq comparative expression across different tissues and these landrace and wild samples, as well as through statistical filtering, we have individual ancestry informative markers, which are nucleotides that are implicated in the transition from wild to domesticated sunflower. And so looking at fatty acid metabolism specifically and filtering through this data set, when looking at Go analysis, we see a lot of highlighting of different metabolic processes and fatty acid metabolism. And when looking at depicted fatty acid metabolism signaling cascade, we find that many of these genes have a strong representation of these ancestry informative markers or the nucleotides that we would want to look at as being causative for domestication, as indicated by these numbers and stars, with certain genes having an exorbitant number of these up in a very high amount. And then finally, we have certain genes in these pathways that have shown strong expression changes, shifting either towards the land races or away from the wild type Nebraska samples that are the multi-branched flowers. And so we can look at these different expression patterns and see what we would want to change from with those genes as we go ahead and modify our plants. And so this all comes together to build what I like to think of as a sunflower engineering pipeline, where we have overall comparative genomics, where we can pick the traits we want and the genes of interest, put those through different engineering design schema, begin reagent testing, and eventually end up with our transformed plant lines of interest. And so applying this to sunflower, we should be able to take this and apply and make interesting new cultivars for use, but eventually going and expanding to a wide variety of other plant species for human consumption. And so I'll end with acknowledgements for different members. All of the tomato, potato, and tobacco work was done in my PhD lab with Dan Voitas. Specifically, Michael and Macy pioneered a lot of the direct injection work, both in Nicotiana and potato. And now the team that in the Blackman lab went through and parsed down that entire candidate gene set that I'm now looking through so that we have good candidates for further engineering. And with that, I'll take any questions.
Very nice talk, Dr. Nasty. I had a question with the fatty acid metabolism before, like with those genes that you found as candidates for altering it. Would you just test it in like Arabidopsis or something that's slightly easier to transform?
For some of these genes, we have knowledge of how you can, whether knockouts or knockdowns can impact the flux through these different pathways. What would be more interesting would be taking, I guess we wouldn't alter Arabidopsis's metabolism. We would go ahead and maybe take a variant from sunflower, test it in comparison to the wild type sunflower in, say, Arabidopsis or something like that, and see how that would change metabolism in that setting.
Thanks. Hi, Ryan. Great talk. Thanks again.
Thank you.
For tracking your transformation efficiencies and the work you've been doing using a couple of markers like Ruby and PDS, those are hard to track in the heritability counts. So is that a problem? Are you developing something new? How do you tackle that?
We've tested multiple different reporters. Right now, why we're using Ruby is it's really easy to see initially, whereas something like luciferase is more invasive. You have to actually add the substrate and everything like that. So you could, in theory, switch to something like that when you're tracking seeds, where it's much easier to, in say the next generation progeny, score and deliver the substrate to those. But Ruby's really nice for initial testing, and why we switched to the Cy1 gene in tomatoes was exactly that reasoning, because when we got really nice shoots in Nicotiana that were PDS positive, or negative, I guess, we were able to track that, but the progeny were infertile, basically. And so we switched to the yellow fruit coloration, which has no impacts on real growth, but you can see the visual phenotype when the fruits are formed. And so those are two ways we've kind of gone about it.
It is a very important crop for the state of California, as over 80% of lettuce is grown here, and over half of that comes from just one county, which is Monterey County, and that is the county where I am originally from. In addition to that, we now have a lot of genomic resources available for lettuce, which provides valuable sequence information for the target genes that we may want to edit for lettuce improvement. Here is an overview of genome editing efforts in the Mitchell-Moore lab, and we have a number of different gene targets. For now, our genome editing efforts depend on tissue culture transformation and regeneration, but we are also pursuing alternative delivery methods such as viral delivery. And for my talk today, I'm going to focus some research on some of my recent research in this area, and my talk is going to be in 3 parts. First, I'm going to talk about a new visual assay for assessing genome editing efficiency. Then I'm going to discuss what I've learned about different promoters for expressing Cas9 and other transgenes in lettuce. And finally, I'm going to just mention very briefly some other strategies for improving the production of edited lettuce plants. So visual screening for CRISPR editing, we've already been introduced to this in the symposium, so I don't have to spend too much time on this, but it is a really valuable tool for being able to rapidly compare different CRISPR editing designs. A commonly used gene is phytoene desaturase, which we already heard something about, that causes an albino phenotype when knocked out, but this phenotype is quite detrimental to the plant. Another strategy is to use fluorescent transgenes, which would then show a loss of fluorescence when there was a knockout mutation induced. But transgenes are also subject to silencing, and so loss of fluorescence may not always be indicative of editing. And for that reason, I wanted to turn to develop a better visual marker for genome editing in lettuce. And I thought that the anthocyanidin synthase gene would be a good candidate. It is responsible for converting colorless leucocyanidins into anthocyanidins, which are then modified to produce anthocyanins that create red pigmented lettuce. And evidence that it would be a good candidate came from a study that was done by our lab in collaboration with Ilya Raskin's lab, which found that when there are transposons inserted in the 5' UTR of the ANS gene, it leads to a reduction in the transcript level and a corresponding change in the phenotype from a very red, deep red color into a green color. And I thought that would be a good gene to target and look for a change from red to green plants. So I used a strategy that we've already been introduced to with I multiplexed 4 different guides, and I put that together with a Cas9 that was being driven by the Arabidopsis ubiquitin promoter, and I put this into a red leaf cultivar called Merlot, and then I could monitor the phenotypes of the regenerating plants even during tissue culture, so it's a very rapid assay. And then I could see how many plants were red and green and see if there would be an increase in the number of green plants, and that is indeed what I found. So wild-type Merlot, as it's regenerating, the majority of the plants are red, but over half of the plants that had been transformed with the ANS editing construct were green. Now, of course, I had to then confirm that the green phenotypes arose through the process of editing, so I did some Illumina amplicon sequencing of the target regions in the ANS gene, and I found that that was indeed the case. So for plants that were green, there were multiple alleles across 3 of the 4 target sites, whereas there was only very minimal editing in plants that were red or had a mixed red and green phenotype. And I think that this is a nice approach because the phenotypes are readily visible in tissue culture and could probably also be applied to any red leaf species. Now, this experiment used the Arabidopsis ubiquitin promoter, but what if there are better promoters out there? And that brings me to the next section of my talk. So we know that in lettuce, the 35S promoter, which is commonly used in other plants, is rapidly silenced and is very unstable. And for that reason, we've been using ubiquitin promoters from other plant species, although we don't really know that much about them. And I wanted to investigate that a little bit further. And I took advantage of the Ruby marker, which we've already been introduced to. This is actually a combination of 3 different genes that act in concert to convert tyrosine to another red pigment, beta-line, so different from the anthocyanins that I was talking about with Merlot. And beta-lines are responsible for the red pigmentation of beets. And so this marker has the advantage of that you can monitor it during the growth of the plant without having to sample the plants or put them under the microscope. And it really gives like a whole plant level readout of the promoter expression. And in Arabidopsis, where the 35S promoter is not as finicky, you can see that the whole plant is red. And I will have to say that I have really fallen in love with this marker. And sometimes I even think it loves me back. And for that reason, I decided to use it to embark on this long-term promoter study. And this is just an overall study design. So I chose to look at the 35S promoter, which I mentioned earlier. We know it's subject to silencing in lettuce, so I would expect to see some differences in the stability of the red phenotype. And then the Arabidopsis ubiquitin promoter, because we've used it very commonly in our transgenic lettuce. And finally, the lettuce ubiquitin promoter, because we had some evidence from the published literature as well as some preliminary experiments in our lab suggesting that it was a strong promoter. And similarly, I introduced these transgenes into plants during tissue culture, and then I can monitor the red and green phenotypes. So unlike the last case where I was looking for red plants to turn green, I'm now looking for green plants to turn red. And then I can monitor them during the first generation as well as in subsequent generations to see if the phenotypes are maintained. I did this in 2 different backgrounds to see if there were background-dependent effects. So right now, all of the plants are currently in the first generation. So those are the results I'm going to present today. And what I would like to show you is that I could see the ruby phenotype already very apparently in early stages of tissue culture. So in calli and shoots, the 35S promoter is, as expected, not producing a high frequency of calli or shoots with a red phenotype. And the Arabidopsis ubiquitin promoter is somewhere in between. And the lettuce ubiquitin promoter has a very strong expression. And although there are subtle differences between the two different background cultivars, the promoters show the same trend for both cultivars. I also looked at the plants throughout development as they grew to larger vegetative stage, reproductive stage, and even flowering. And I was happy to see that the flowers also express the very strong ruby. And I will mention that red leaf cultivars such as Merlot, those flowers are still yellow. So this is a really interesting thing to see. I did see red expression also in roots, and it doesn't really appear to be expressed in the pollen. So this suggests that the lettuce ubiquitin promoter is a really good one for driving transgenes, and that is what we're going to move on to do. I will say that the mixed phenotypes are also really interesting. So I think this tells you a lot about what is going on during regeneration. You think you know what is happening, but you really don't. And this is, I think, evidence of that. So I'm hoping that this collection will provide a lot of study material and will help us understand what is happening to promoters as they're going through this process. Lastly, I'm going to touch on additional strategies for producing edited lettuce plants. So now that I have this really easy screen, I can test many different construct designs. I already mentioned the lettuce ubiquitin promoter. If I use that to drive Cas9, I can see if it will actually lead to more editing. But one thing I haven't talked about yet much is the guides. But luckily, Brian in his talk was already mentioning this, that you have a variable efficiency in the guides. So some guides work very well, some guides don't work at all. And you can you can just include lots of guides to sort of buffer yourself against guides that may not work as well. But it would be good to have some knowledge ahead of time of which guides were good. And Brian showed that you can do some implanted testing. You can also do in vitro testing. And then there are also some guide prediction algorithms that can be used to predict whether it would be a good guide or not. Testing, even if it's a fast platform, does require time. And the algorithm prediction methods, I will say, do not really perform well, at least in our experience. And I think Brian mentioned that too. So there are many different tools that are available for guide design, but very few of them give any consideration to plants at all. And when they do, it's only to provide plant genomes as a screening for off targets. And they still use algorithms that are developed for animals. And you can see that for lettuce, there is no correlation between these guide activity scores that are predicted and the observed editing efficiency. And from this, I conclude that plants are not animals, and that we need better algorithms for predicting guide success in plants. And currently, I am collecting, because of course for that, we need a lot of data. So I'm assembling a data set from the published literature. But of course, I would like more data, especially data that doesn't end up in publications, usually for those guides that don't work. And I'm looking to collaborate with anybody who is also interested in this question and may have data to share. I want to use a machine learning approach that training on the plant data set to see if it will produce better predictions for guides and plants. And with that, I'm going to end my talk and thank the people involved. The people with stars by their names are actually students, undergraduate students, and one high school student, and a number of other people, of course, in our lab, including Richard Mitchell-Moore for providing a great place to work. So thank you.
Hi.
Hi, Juan.
Yes, I have a question regarding the mixed phenotype in the certified Ruby.
Yeah.
Have you tried to genotype different part of the leaf to see if you are getting a chimeric plant or silencing?
Yeah, so actually that is something that's going to be happening like over the long term, because of course I think that the mixed phenotypes can arise through several different processes. So it could be incomplete integration events. It could be subsequent recombination after transgene integration. It could be due to silencing. And so we are definitely going to be doing a lot of genotyping of this material, just haven't gotten there yet.
Hi, Beth. Thank you. Very intriguing talk. Is protoplast transformation and regeneration a thing in lettuce, and would you leverage it for some of your early studies?
Yes, okay, so protoplasts are something that other researchers in the lab had been interested in doing. I wouldn't say that we are not totally uninterested now, but we had sometimes very successful protoplast production and transformation, and it seemed to be quite variable and very finicky. And so far, we don't have good methods for regenerating from protoplasts, but I do think that this is all possible. I don't think that it is never going to be not a thing for lettuce. We think we need more people working on it to pursue it.
So we find in corn, soy, all of our row crops with different guide RNAs to have about a 95% success rate. I noticed in the lettuce numbers you had up there that was a fair bit lower. I wonder how much of that is also tied to expression systems, right? If you can express things efficiently enough, all of our systems are very high expressing, very efficient. Somehow that can maybe overcome some of the variability. Kind of wondering what your thoughts might be there.
I really like that question because I do wonder about how much, so if you change your expression system, can that turn like a bad guide into a good guide? Or even having a better Cas9, like if you're getting better, maybe if you can get by with worse cutting in some cases versus other cases. And so I think that there can be elements of the design that can change whether a good guide is a bad guide. I do worry that this makes it hard to train algorithms. And but I'm hoping that with enough data, even if there's a lot of noise due to those factors, that we can find some kind of signal in there. I'm also kind of curious to know, because there's a lot of people here, if there's if their experience is that their guides are just generally more successful and this is not a problem. And I'd be wondering to know which species are kind of more problematic.
Thank you so much. Great, thanks everyone. Before we go to lunch, I have a couple points to make. So first of all, restroom is on this side. There are also restrooms downstairs at the same location. Secondly, after you grab your lunch, you can go outside of the glass door. We have a lot of seating outside. There are a lot of stairs, a lot of tables. Also, there are a lot of tables downstairs as well outside. And lastly, and most importantly, the lunch is going to be serving from this door. So if you can line up through that door, it'll be great. You will get utensils and plates first, and then go all the way to get your food. Thank you. And we will be back at 1:25 for the afternoon session. Hello. Hello, everyone. We're gonna start our afternoon talk very soon. First of all, my name is Han. I'm a fourth-year graduate student at the Williams Lab at UC Berkeley, and I will be introducing Dr. Juan De Bernardi. So Dr. Juan De Bernardi is a plant molecular and development biologist. He is associated with the laboratory of Professor Dobkovsky at UC Davis. Initially as a postdoc fellow of the Human Frontier Science Program, and later as a research specialist of the HHMI, Dr. De Bernardi's work is focused on understanding the genetic networks controlling wheat plant architecture and their effect on yield. He is also a project scientist at UC Davis Plant Transformation Facility and leading projects to develop technologies that improve crop transformation and genome editing. So welcome, Dr. De Bernardi.
Okay.
Sorry, here I'm seeing the next presentation. That's okay? Yes. So, thank you very much for the invitation. I'm really excited to present here in Berkeley. So this is probably one of my first presentations since COVID. So I'm very excited about that. So I have divided the presentation in two parts. In the first part, I will talk about technology or method that we developed. It involved work that I did during my PhD in Argentina in Javier Palatni's lab. And I continue here in Jorge Dukowski lab and in collaboration with David Tripoli in the transformation facility. So I will explain more or less that transformation or plant regeneration technology. And in the second part, I would like to present a simple example, simple example of what we can do right now in wheat with the technology that we have developed. Okay. So I will start with this very simple slide that summarizes one of my main motivations on plant research, that is to translate the ability to translate our knowledge into crop improvement. So that was one of the main reasons I came to Davis to do my postdoc in Jorge Dukowski lab, that is a wheat lab, to translate part of my knowledge in molecular biology. I did a PhD in Arabidopsis, so I wanted to work in a crop. And I think that we all agree that gene editing is a great, amazing tool to achieve that, to translate our knowledge into crop improvement. But when I came here, I realized very quickly that I was very naive, because we can introduce modifications into the cell, but to regenerate a whole plant from the cell that had a modification that we want, that's a very difficult problem. And it was the main bottleneck, currently is the main bottleneck on applying gene editing to improve crop improvement. So for most of the crops, here I'm showing a typical transformation that we have at the UC Davis plant transformation facility with transformation. You can see the efficiency is very low. So for many crops, the efficiency of transformation is very low. Only a few genotypes can be transformed. And the protocols are very labor intensive and require a lot of space to supply the tissue that you use for transformation. So that was kind of, for me, coming from Arabidopsis, that I can do floral dip. That was a big shock, so I was very limited to do research or trying to translate my knowledge into wheat. So, but the problem of, it's not moving. What should I do? We close that. There it is. Thank you. Okay, so but the good thing is that what I call the regeneration process or regeneration problem, many groups have been working on that process, and now we have a better understanding of the process. So normally, what you want is to take a somatic cell, and the process involves the differentiation of that somatic cell. It involves cell proliferation to generate a cali, and from that cali, then you had an acquisition of new phase that will give up to meristem, should apical meristem, or root meristem. So the knowledge that we have on that system allowed to use to test many developmental regulators to try to improve the system. And here there is a list of all of them. Today, all different presentations talk about the baby boom bushel system that I think is a great example of how using developmental regulator you can accelerate or improve that process that plants normally have, but is at low efficiency. But something that is very common for all this system is that when you use meristematic or embryogenic genes to induce regeneration, if you continue expressing those genes later on development, they introduce a lot of developmental defects that can affect fertility or plant fitness. So The thing is that something very nice that did Corteva was to develop a lot of different strategies to control and fine-tune the expression of these developmental regulators in order that you can boost regeneration and at the same time get fertile plant. So today I'm going to present our technology that use a different kind of genes that allowed also to improve regeneration. And what was nice, or what they consider is very nice of this system, is that it doesn't introduce developmental effect, so it's easier to use. So first, now I will provide a little bit of background to explain how we arrived to this technology that we call GRF chip. So that involved work that I started at my PhD in Argentina. So I was interested in characterizing this family of transcription factor that is called growth regulating factor, or GRF. So they were cloned and discovered by the group of Hans Kendi in Michigan many years ago. So this is a plant-specific transcription factor family. They are very conserved, and they are defined by the presence of these two domains. The WRC that mediate DNA binding, and the QLQ that mediate protein-protein interaction with another gene family called GRF interacting family, or SHIF. So the SHIF family is smaller. It doesn't have the protein here, or the gene here, doesn't have a DNA binding domain. And the domain that they had, the SNH domain, mediate interaction with the GRF to make a complex, and also with chromatin remodeling complex. So that way, this function as a DNA binding, and GF can recruit chromatin remodeling complex to the targets. The GRF also is target of a microRNA, that is microRNA 396. And during my PhD, I worked a lot on studying this family. One of the questions that we wanted to know is what is the role of the GRF in plant development. So one of the first things that we did, and we observed, is that the GRF has an expression pattern that correlates very well with a cyclin reporter. So this is a cyclin B gas reporter. So you can see that the expression pattern is very similar. But we also noticed that this expression pattern of the GRF is mediated by the activity of the microRNA. So this is the expression pattern of microRNA 396. It's completely opposite. And here is a reporter where we remove the microRNA binding site on the GRF. So you can see that if you remove that regulation, the GRF has a constitutive pattern. So this is clearly mediated by the microRNA and the transcriptional activity of the promoter. So we next ask, what is the role of these genes in vivo? And for that, we generated a transgenic plant that overexpressed the 396. So this should be considered as a downregulation of the GRF. And you can see that the plants are smaller. And the same phenotype can observe if you make a knockout on the SHIF, the interactor of the GRF. So clearly, these genes are involved in controlling plant organ growth. And we also cross this plant with the same reporter that I said before, it's a cyclin reporter, and we observed that there was a reduction in the expression of the reporter, a reduction in cell proliferation. So at the end, the conclusion from here is that these genes control organ site, and they do that by controlling cell proliferation. But we also observe that the effect of the controlling cell proliferation is not only important to control organ site, but at some point, if you have a strong downregulation of the complex, like if you cross these two, you affect meristem homeostasis. So the plant doesn't have a meristem. So cell proliferation is important to organ size and meristem homeostasis. The opposite, if you overexpress the GRF, you can get higher plants using constitutive promoter or removing the microRNA, or having the two genes expressed together, GRF plus SHIF, even increase even more the organ size. So to summarize this, at the end, we ended up with this kind of model where we have GRF SHIF that control meristem homeostasis and plant growth, and the activity of the complex is controlled at multiple levels: transcriptional regulation, at post-transcriptional regulation by the microRNA, and then we also start seeing a lot of evidence that there were competitive interactors that will affect the assembly of the complex. So from this evidence that we have, we got the idea several years ago to make this construct where we put GRF and SHIF in the same transcript, so they will transcribe as a single polypeptide.
So the chimera, what we call this chimera. So, but this new protein, since they are expressed together, linked by a polypeptide, they will form the complex more efficiently without any competition. We test that in Arabidopsis, and actually we were able to see that when we express this as a chimera, we can boost the activity of GRF. So basically, we have a simple tool to boost cell proliferation in planta. Javier and I had the idea of testing this in wheat. Okay, what happens if we increase cell proliferation in wheat? What will be the phenotype? So first, we decided to select the best GRF to make and chief to make our chimera. So wheat has many GRF and chief. So we check expression, and you can see that like in Arabidopsis, most of the GRF has similar expression pattern as cell cycle associated gene. So they are expressed in cell cycling in organ that are under cell proliferation. So from here we select GRF4 and Chief1 as our main our initial candidate to make the chimera. So we transform this in wheat, and to our surprise, I mean, we got a huge increase in regeneration efficiency. I say surprise because, as I told you, we were expecting some effect on cell proliferation and maybe some improvement in regeneration, because there are a lot of evidence that connects cell proliferation with regeneration, but we were not expecting to see that big increase, that boost in regeneration. So that increase is very robust. We could see over many experiments. Right now, we have seen this many times, but I mean, we are super confident with that. But not only we observed increase in regeneration and transformation efficiency, we also observed that the whole process of transformation goes faster. We reduce almost to half the time that you require from inoculation with the agro to get the plant. And also, wheat transformation is very hard and very difficult because the quality of the explant affects a lot the efficiency of the system in the traditional methods. With GRF chief, we observe that we have less constraints in the quality of our plants. So now we can grow our plants in regular growing chambers, and we can get a good efficiency of transformation. The other point that was very interesting is like the plants that we recovered from here, they were completely fertile. So we don't have to remove or control the expression of those genes. We were using maize ubiquitin promoter, and the plant were just fine. So that is very simple and easy to use. And the first thing that we did was to combine this with gene editing. So we put in the same tDNA our cassette expressing the GRF shift with Cas9. We target a gene that in wheat produces a very clear phenotype, that is the name is gene Q, and the phenotype is increased floret number. The first thing is that we transformed this, and we were able to still maintain the high efficiency of transformation. So we knew that the more complex DNA was functional. But what's very interesting too was we were able to recover a high frequency of editing plants. So here it was around 100% of editing with this guy. The other thing that was very interesting for us, that is very interesting with this system, is that we could expand the genotype that we can transform. All the data that I showed before was done in Kronos, that is a durum wheat, a tetraploid wheat, that is our lab system. But we were able to transform Deserkin, that is a UC Davis breeding line, is the most important breeding line in the program now. We were able to transform some recalcitrant hexaploid wheats, and we were also able to transform a triticale line that before was recalcitrant from our breeding program. So as a summary of this first part, I can say that GRF shift increased efficiency, the plant are fertile, we have a high frequency of editing events, and we expand the number of genotypes that we can transform. So in the second part now, I want to tell you a simple example where these two things, high frequency of editing event and the expansion of genotype, how we use that, or how that changed the way we are doing research now in Davis on wheat. And the example that I will use is a project where we are trying to generate novel genetic resource to control plant height in wheat. So this project starts in conversation that I have with our wheat breeder, Josh, in the field. So Josh, he was showing me triticale plants. So triticale is a hybrid between wheat and rye. So it has AB genome from wheat and the R genome from rye. It's very interesting, triticale. It's a kind of novel new crop because it combines the things from rye and from wheat, and in Davis, the material that the Josh has really have, for example, the yield is 50% higher than wheat, and this plant also have more than around 40% increase in biomass compared to the best wheat line. So I think that triticale is a very exciting system and crop. But so and here in this picture you can see this is a triticale spike, these two, compared to a wheat spike. So you see the higher yield is because this plant produces are monsters. They produce a spike that are super big. But talking with Josh, Josh told me that these big spikes are too heavy, and the plant had lodging problem. So basically, what he told me is, I would like to make this plant shorter. So that was a great challenge for me, and I took this idea to develop new plant high alleles. Why I'm saying new? Because during the green revolution, I think everybody knows here that in wheat, breeders select for dominant mutation in DELA genes. So these dominant mutations, DELA genes is involved in GA synthesis. So they select for mutations that impair the GA binding domain. So basically, GA cannot repress the DELA activity, and as a result, you have a dwarf plant. So these are all the series of alleles that were selected in different breeding programs. You can see it provides very nice control on plant height. The problem that you have with this DELA dominant allele is that they have pleiotropic effect. So they not only make the plant shorter, they make all organs shorter. So for example, here I'm comparing the wild type and the RHT1B seedlings, and you can see that it makes the seedling shorter. And that's a big problem for people that want to plant wheat in semi-arid region, because they want to plant deeper in the soil to get the seed close to the humidity. But to plant deeper, you need seedling that can reach the top. So they cannot use RHT1 as a plant high allele. So there is a necessity to develop new. So again, the problem was to develop novel high alleles and avoid DELA genes to do that. I had the idea that approach plant high in wheat in a different way. I consider plant high as it's the stem elongation, basically, the peduncular elongation that take part during the reproductive stage. So instead of targeting genes that control plant high or plant size in general, I was focused. I wanted to target regulator of the flowering transition. And I have a good candidate for that because I spent a lot of time working with this model, microRNA 172, AP2-like transcription factors. So the microRNA 172 repress the expression at the post-transcriptional level of AP2 transcription factors. And I have evidence that this system is involved in reproductive transition. You can see the microRNA is very specifically expressed in reproductive organs, the spikes. Also, using a transgenic approach to inactivate the microRNA 172, I was able to get shorter plants. So I say, okay, these microRNAs express reproductive tissue. If I inactivate, the plants are shorter. And also, I have this evidence in Jorge's lab, we have a Tilling population, EMS mutation. I found one mutation, point mutation in the microRNA binding site of one of the AP2 transcription factor that affect the microRNA interaction and make the plant a little bit shorter. So the idea was, can we target the microRNA binding site using CRISPR-Cas9 and generate more, probably stronger alleles? Will those alleles affect seedling development? Can we reduce plant height independently of the DELA alleles? And can we translate these alleles into an elite cultivar? So before, to target all these questions was, I would say, impossible. But now I think we are in a good position. So the first thing we did was to transform and target the microRNA binding site of one of these AP2 transcription factors in Kronos. Kronos already had the RHTV1B, the semi-dwarf allele. So we got, in the first generation, around, here are 43 independent events. We got the plant, and we classified the plant in wild type. We sequenced the plant, we classified the plant in wild types, heterozygote for mutation in the microRNA binding site, or plant that were homozygote or allelic with mutation in the microRNA. Just from this data in T0, this experiment was very fast. It took like maybe 4 months to get the result. We were able to see that we could get a stronger reduction in plant height as we were expecting. Here is, before I say 5 centimeters, here is around 15 centimeters. The next question that we asked was, okay, would them affect seedling development? We select wild type plants and homozygote allelic plant, and we grow the seedling, and there was no effect as we were predicting. So the seedling size was exactly the same as the wild type. So the plant height was affected, but not the seedling. The next question was, can we reduce plant height independently of the DELA? So remember, in Kronos, we had the RHT1B allele. But also in the lab, we have another Kronos material that is an ER isogenic line where we introgress the wild type allele, RHTV1 allele. Again, we transformed, we got around 45 independent events, and we checked plant height, and again, we were able to see a very strong reduction in plant height in this genotype. What is very interesting is that you can see if we can compare the edited lines with Kronos having the RHT, the DELA, the reduction in plant height is almost similar. A little bit less, but it's similar. And when you check the seedling, you can see that the RHTV1, V1B reduced seedling size, but the CRISPR lines have the same the same length. Okay, so basically with this result, we can say that yes, we can reduce plant height independently of the DELA, and we are not going to affect seedling development with this CRISPR line. And finally, the last question was, can we incorporate or can we transfer these kind of alleles into elite cultivars? So, and going back to the first question, talking with Josh, I wanted to edit this site in Triticale to solve Josh's problem. So with the technology, we were able to transform Triticale, and Genome BNR of Triticale has a pump site next to the target site, so we CRISPR that. And we were able to recover lines that have a very clear reduction in plant height and no effect on the seedling. So we were very excited about this result. But something that you can notice also from this slide is that we not only reduce plant height, but also we got plant with different height. So these are two independent lines. When we sequence those lines, we notice that in the B genome, they have the same mutation. It's a deletion of one nucleotide. But in the R genome, they have different mutations. One has an insertion and one has a deletion. When you check the interaction of the microRNA binding site with these two different microRNA binding sites, so this mutation is weaker than this one. So you can see the reduction in the energy. And that's correlated with the height. So a stronger allele that has a stronger effect on microRNA activity will produce an allele that is stronger and will reduce more the plant height. So I say, okay, this system seems to be very quantitative in controlling plant heights, not just qualitative on offer, it's very quantitative. So what we did is we backcross this line and we generate more alleles, and basically what we got is this. So we got a huge, here I'm showing a few combinations, but we have a large combination of different mutations where we can control plant height just doing editing in this specific site at the centimeter resolution. Just for example, this one has a single, we can do that in different way by selecting the identity of the mutation that we introduced, and also playing with the dosage of mutation, because we had 3 genomes, we can play with the number of genomes that we mutate. So here you have, for example, a plus mutation, a deletion, plus and deletion, and we have a stronger deletion that makes the plant even shorter. And if you wonder how is the seedling size of this, no effect on seedling size. So basically, we were able to control plant height without any pleiotropic effect on seedling state. So that's the Okay, so to summarize and conclude the presentation, so this is what I showed before. And I think with this example, I try to show that how CRF shift combined with CRISPR-Cas9, in this case could be any editing tool, can produce rapidly mutation to test different hypotheses, in this case in the crop, and how, for example, in this case, we edit AP2, and those alleles can be useful source of novel high alleles with less pleiotropic effect on plant development. Okay, so I would like to thank a lot to Javier, that was my PI in Argentina, Jorge, when I came here to UC Davis, Chao Song, he helped me, or he's leading now the project on Triticale with all the editing result, and shows that we are planning to do a field experiment with these plants. And I would like to especially thank David Tricoli, that is the managing director of the transformation facility. I was very lucky to meet him in Davis. It changed my career completely. So now I'm like a plant transformation guy. So I'm very happy, I'm very thankful of David. Okay.
Hi, thank you. Great talk. I have 2 questions about the microRNA 172 mutants. Or the mutants that are related to that. How much earlier do they flower, and do they have reduced vegetative biomass because of their early flowering?
No, that's a good question. Yeah. In wheat, there are 4 AP2 targets of this microRNA. Compared to Arabidopsis, the functionality of this AP2 is more. There is less overlap so you have AP2 that are more related to flowering time control. And so I select this AP2 because I knew already that it's less pleiotropic on this other phenotype, like flowering time. My interpretation is like a microRNA 172 is I don't like the word master regulator, but let's say master regulator of the flowering transition or reproductive transition. And that involves many, like flowering time, and in this case, it's the peduncle elongation, so yes. But this AP2, if I target other AP2 that I know, I will affect flowering time, or gene Q that I mentioned in the presentation is one of the main genes on wheat domestication that control threshing. So I will make the plant shorter, but the grains will fall on the field. So this AP2 is a different one, and it's less pleiotropic.
Hi, thank you. I was wondering if you can use the GRF system to generate non-transgenic plants, because I'm imagining you have a transgenic plant to facilitate like the transformation, right?
Yes, that's a great question too. For example, in monocots, I was very interested on that question in sugarcane, for example, because sugarcane, you want to transform it, have edited, but it's better not to have integration, because if you had to segregate out, it's a mess. In wheat, we don't have that problem. But I test I did a transformation experiment without selection and regenerating plants without selection, and then I screen for editing and insertions. And I was able to recover around 5% of plants with edit and no insertion using this system. I haven't done that. If you want, I'm not very good at protein. I'm not a good biochemistry. If someone wants to do that, yeah, it would be great to do RNPs with. So there is a paper from that the Kaisha Gao group, they transform RNPs but with DNA encoding for the chimera. So they did transient in that way. But it would be great if we can do protein. Yeah.
one question from Zoom. It's asking about the process of generating the transgenic plants. So the question is, did you first generate GRF4, GIF1 transgenics and then perform gene editing, or do you do it together?
They are together in the same tDNA. I don't know how much time I have. So, because one idea that we have also is related to that, but not very related. So it's what happens if you want to transform other stuff that is not chain editing. In chain editing, they go together in the same tDNA. You can segregate the whole thing out, and it's perfect. But let's say that you want to make a transgenic to do a reporter. So you cannot put the CRF shift in the same tDNA, because even if the plant doesn't have developmental effect, you have something there that may affect your reporter. So I don't like that. We try to retransform plants that have CRF shift inside, but that method didn't work very well because we were getting very large calli, but it had problem to regenerate. What we are doing, and it's working well with us, is something similar that Corteva is doing with bushel with the altruistic method. So we are doing co-transformation with two agros, one with the vector that has your reporter gene, whatever, another agro that has the chimera. We co-transform and we can recover plant in that way. Sorry,
So our next speaker is Dr. Yi Jing. Yi Jing is a director at Research and Development at Inari, the seed.
Here. Welcome back, and now we have 3 more talks to go. And first one is from Matthew Davis. He's a PhD candidate at UC Davis, and he received his undergrad from University of Florida in majoring in biotechnology and a minor in entomology. And now he's pursuing his PhD in plant biology at UC Davis, studying genomics of walnut and pistachio. Matt is specifically interested in studying long-term effects of tissue culture on especially orchard crops and their genomics, and he would like to use genomic tools to help or assist climate adaptation in perennial crops. Please.
Hey everybody, can you hear me all right? All right, awesome. So first I want to start with thanks to the organizers for inviting me and allowing me to come and speak, and preemptive thanks to you guys for listening for the next 15 minutes. And I kind of want to pivot a little bit. So we've been focusing a lot on gene editing, of course, but I want to talk a little bit more in depth about the material that we're using for gene editing. And so what you guys can see right here in this bottom left corner is a plate of repetitively embryogenic walnut. So these walnut embryos were introduced into culture in 1995, so it's been 27 years, and they've been kept in culture ever since. Now in the background, you can see is a picture of walnut cultivar Chandler. That tree specifically is our reference genome, but additionally, it's also a cutting from the tree that these walnuts arose from 30 years ago. So we have a really cool opportunity to look into mutation in these repetitively embryogenic cultures. So first, I did show my hand a little bit, but I do want a little tiny bit of audience participation. So I'm sure if everybody could just raise your hand if you consider off targets when you think of gene editing. Where's my hand too? Okay, cool. So probably all of us. Now, who considers the de novo mutation that's occurring in tissue culture in their products? Okay, so a lot of us. This worked out really well for me, so I'm preaching to the choir, and I think things are gonna go well. I don't have to convince you of too much. But this is kind of setting the stage for a phenomenon that was described by Dr. Barbara McClintock. And so in this quote here, you can see she says in her 1983 Nobel lecture, she says, "No two of the callus-derived plants are exactly alike, and none is just like the plant that donated the cell or cells for the tissue culture." And so what she's really getting at right here is this idea of somoclonal variation. And what somoclonal variation is describing is differences that we see arising in plant tissue from the same genetic material. And so if we look at this little cartoon diagram right here, we can say that cutting from this plant was introduced into tissue culture. And then as it was propagated over time, we start to see these phenotypic differences over here. And this is all arising from the same original genetic material. Now, this obviously is a huge problem for plant breeding and for gene editing, because we want the product that we're editing to be the same product when it goes out into the field. But due to somoclonal variation, this occasionally cannot be the case. And callus has been seen to contribute to somoclonal variation. And so what we see here, this chart is actually a little counterintuitive, so I'm just going to walk through it. The lower bar is higher mutation. So this chart's on the negative log scale. And so what we're seeing in this red is we're seeing rice callus, and its mutation rate is about 2 orders of magnitude higher than the same rice grown out in the field. Now this is mutation rate per base pair per year. And then at the end, since we're looking at trees, I want to look at those 3 species over there. So we're seeing peach, eucalyptus, and poplar. We see a relatively low mutation rate compared to our short-lived plants that we're seeing up here in our gold and green, rice and Arabidopsis. And so when we start to think about these embryogenic walnut cultures, we're considering not only the callus mutation rate, but also the mutation rate that we're seeing in trees that tends to be lower. And so when we start to talk about these embryogenic cultures, first it's a good idea to get a handle on where they come from. So like I mentioned previously, so this guy, Chuck Leslie, you can see him right here, went out in 1995 and took a cutting from, or took some tissue samples from this tree. He introduced them into liquid culture, and he derived direct embryos, directly embryogenic walnuts. So essentially what that is that's an embryo arising directly from tissue without a callus phase. And now these direct embryogenic walnuts are also repetitively embryogenic. So they're continuing to give rise to new embryos. And they've been doing that for the last 27 years. Now, about a year later, in 1996, a cutting from this tree that produced these direct embryogenic walnuts was taken, planted out in the field, and then became our reference genome. So we have this high quality genome of this tree that was grown out in the field for about as long as these embryos have been in the lab. So it gives us a really cool opportunity to start comparing things. How do I go back? Oh, I figured it out, sorry. All right, so as we're thinking of gene editing and these embryogenic walnuts, they gave us kind of an idea. We started to think, what could be going on in these walnuts? They've been in culture for so long. They're repetitively embryogenic, so there's something funky about them. And they could be a really, really useful tool for gene editing because we're producing embryos straight from another embryo. And so when we look at the mutation rate and we think about this somoclonal variation that we talked about a little earlier, we were wondering, is there a high level of mutation in these repetitively embryogenic cultures? And if so, where is this mutation occurring? So the first thing we had to do to figure this out is extract some DNA. So you're going to see this CR2 moniker up here. So we're going to see CR21 and CR22. CR2 is just our embryogenic lineage name. And we're going to have it's essentially two individuals. So CR21 is one individual, CR22 is another individual. Now, because we're working with trees, we know that they're highly heterozygous. And so we lucked out once again because we had 2 biological replicates of Illumina reeds from the same tree. And we can use these reeds to correct for any of that ancestral heterozygosity that's not accounted for in our reference genome. So we extract DNA on our embryos, and then we sequence with 150 base pair paired end reads, and we already had the sequence data for 100 base pair paired end reads. So we're doing whole genome Illumina sequencing here. And then afterwards, we want to call variants on both, and we want to compare. And so to compare, the first thing that we wanted to do was set our 2 individuals equal to each other. So our replicates from our reference genome, or from our reference genome tree, and our replicates from our culture individuals, and that's to avoid any mutation that arose in just one branch of the tree or just one cluster of embryos. And it's also to account for any sequencing error, because the odds that a sequencing error happened twice in the same exact location is fairly low. After that, we only wanted sites that differed between the two, because that's where our mutation will lie. And we had some errors with our haplotype caller calling things that didn't look like heterozygous sites as heterozygous. So we used a chi-square test on our alternate and reference read depths to filter out anything that didn't look heterozygous. And we can talk more about that later if you're interested. And we ended up with over 4,700 variants. Now, we wanted to define our mutation as something that we could be pretty confident in. And so we only took things that started out in our reference tree as homozygous, and then are now in our embryos as heterozygous. So it's one change on one haplotype. And now that we have all this data, we can look at these mutations in context. And so there's a lot going on this chart, so I'm going to walk through it. Up at the top, we see our single base substitutions. So if we focus in on this blue chunk in the beginning, this is a C to A substitution. Now, on the bottom, we have the context that we're seeing these substitutions in. So this really big spike that we see is a TCT. So that means the C was surrounded by a T upstream and downstream before it turned to an A. And then on the y-axis, we have our count. And so we can see some interesting things, right? Because this substitution seems to occur more than any other C to A substitution. Now, these are counts, so it is possible that this is just something that is extremely common in the genome. And that is something that we do want to follow up on. But when we look a little bit further, and so now what we've done is, this is the same exact plot in the middle. This is our embryogenic culture. We've smushed it a little bit, and then on top, we have Arabidopsis, the same single base substitution context plot, and then rice, another context plot on the bottom. We can see that there are some interesting similarities and differences between them. So rice kind of shares a similar pattern to these walnut embryos in that blue section, but Arabidopsis is really, really low. And then we can see our C to Ts are very high, and that's something that we expect because we know that it's a common single base substitution. But then we can see T to Cs are pretty high in our walnut embryos, higher in rice than in Arabidopsis, but still seem to be relatively highest in our walnut embryos. And so we want to look a little bit more into these comparisons and try and find out why we see these differences, and also potentially look at walnut callus and see if we see a similar pattern across the walnut callus that we see in the walnut embryos. Now, if we want to see where these mutations are occurring, so we just looked at all of these mutations in context. Now we're looking at where they're occurring. We can see on our y-axis here, we have mutations per base pair. And on our x-axis, in the green line, we can see this is our gene body, measured in % because genes are all different sizes. And then we have 2,000 bases upstream of it and 2,000 bases downstream of it. The data is a little bit noisy, but we can see that there is a lower level of mutations inside our gene bodies as compared to upstream and downstream. So it raised the question for us of, is this purifying selection? Are we seeing selection in these populations on genes? So in order to answer that, we have to look at our mutations within the coding sequence. So we saw a total of 190 mutations in our coding sequence. 138 of them were missense, 9 of them were nonsense, and 43 were silent mutations. So from this, we want to predict our null DNDS ratio. So what our null DNDS ratio would be there's no selection happening at all. No selection in this embryo culture lineage. The value for that was 2.2. Now, when we see our actual observed DNDS, we see 3.2. The reason that this doesn't suggest purifying selection, because purifying selection would be a lower value than our null. This is a higher value than our null. So there was no purifying selection in these embryo culture lineages. So I'm going to leave you with a little bit of math, but don't worry, I've already done all of it, so you just have to follow along. If we want to figure out what our mutation rate is in our embryogenic cultures, what we do is we take our number of mutations, we put it over the number of years they've been in culture, so 27, and then we put that over the total number of base pairs in our genome. And we end up with 1.44 times 10 to the negative 7 mutations per year. Now, when we look back at that chart that we saw at the very beginning, and remember, this is a little bit funky, it's the opposite. So lower bar means more mutation. we see that it's really comparable to what we're seeing in callus. So what I want to leave you with is maybe it's important that we also consider this de novo mutation that's occurring in callus because we see that there is a much higher level of mutation occurring than we're seeing in field plants. So with that, I want to thank everybody in the Monroe lab for all their support, as well as the Brown lab, and additionally Julian Maloof, John Davis, and Noah Feinberg for the additional help that they gave me. And I'll take any questions that you guys may have.
Hi, I have a question. So the data that you presented, this is all from Illumina sequencing, and I was wondering if you had done any long-read sequencing, because I'm so curious to know if there are structural variations that change in these long-term cultures.
Yeah, that's actually what I'm doing right now. So I have hi-fi data that I'm waiting to analyze, and I'm going to be looking specifically for TEs and structural variants. So yeah, I'm really excited. I think there's going to be some really cool stuff in there.
Nice talk, Matthew.
Thank you.
Quick question. I don't know, have you do comparisons comparing the synonymous mutations versus non-synonymous mutations and see whether there is a bias towards that as well?
Yeah, so how we got the predicted null value, or specifically if there's a bias to a synonymous mutation within the gene body? Yeah, so we haven't done that analysis yet, but I think that's definitely something where we will do because it's a cool idea.
Do you think some of the differences in the types of mutations you observed are related to specific types of DNA damage or DNA repair pathways that are happening?
Yeah, I think there's a lot of funkiness going on in tissue culture in general. But yeah, I think with the context plots, we can see patterns in humans that are indicative of specific types of DNA damage. Now that is in humans, it's not specifically in plants, so I don't know how much we can really extrapolate from that. But if we can extrapolate from it, which again, I don't really think that we 100% can, we can see that there's some like reductive oxygen species damage, like associations as well. So I think that's part of it. But yeah, I think that there definitely is, but other than that, I don't know how much more specific I can get.
We can go for one more last question.
Yeah, great talk. I had a question behind like the reason for this difference in mutation rate, and I was wondering, have you tried to infer, for example, like the number of cell divisions, and if that's correlated with mutation rate? Because I imagine in tissue culture they're dividing and proliferating much more.
Yeah, we've been trying to figure out how we can go about determining number of cell divisions because it has been so long, and we have like a rough estimate of we're actually like separating and propagating them once every 3 to 4 weeks. But further than that, it gets really tricky because each culture kind of divides at a different rate. So right now we're sticking with this, and then we're going to do some comparisons out in the field as well.
Thank you, Matt.
Yeah, thank you.
So our next speaker is Nicolas Carvalhas. He's from IGI next door, and his research focuses on stomatal development in rice. He would like to use gene editing to seek understanding on how rice stomatal density can fine-tune rice to be resilient to the climate changes. And Nicolas is broadly interested in gene editing, and he would like to facilitate and improve the crop varieties. Please. And transcription factors associated with nitrogen metabolism, working with Shivan Brady at UC Davis, and now she's working on monkey flowers, especially Mimulus guttatus adaptation, local adaptation, and then using genomic and genetic tools to uncover regulatory networks to understand how plant growth and development matches to their environment. Ali, please.
Last talk, you guys made it. Okay, so I'm not gonna talk about crops at all. Okay, so my talk today is called Characterizing Mimulus guttatus Adaptation to Serpentine Soil. A lot of these terms are new to you guys. So let's just talk about nutrient availability. So we know that bioavailability of nutrients affects how plants grow and how they develop. And most of what we know about this is either from Arabidopsis or crop species. And all of those plants are really needy. They need a lot of nutrients. And so in my work, I'm interested in understanding plants that adapt to really extreme, harsh environments. And so the environment that I study is provided by serpentine soil. So serpentinite, I have a rock right here, is the state rock of California. And it is derived from the breakdown of ultramafic rocks. And so we are on the crust of the Earth, but sometimes the mantle gets pushed up, usually in areas of high geologic activity. And then there's an exposure of rocks from the Earth's mantle. And so because of this, the chemical composition of these rocks is super different from what we would consider normal soil. So the broad definition of serpentine soil is that there's a really skewed magnesium to calcium ratio, but there's also much lower levels of both macro and micronutrients. So nitrogen, potassium, nitrogen, potassium, phosphorus, calcium, and sodium. There's also high heavy metals, including nickel and cadmium, chromium, iron, and then also high pH. And also these are highly drought prone. So like, yeah, horrible places to live, right? But actually incredibly important for California floristics. So even though serpentine soils only cover about 1.5% of the land in California, 13% of endemic species are now found on these soils. And so they create this really strong selective pressure for being able to survive in this harsh environment. And some species cannot survive on serpentine at all. Some have some populations that can and some that can't. And then some plants are actually restricted to serpentine soil. And so I'm going to tell you a story about plants that have some that can survive and some that can't survive. And so in the Blackman lab, we study local adaptation. And this is where populations from species that are found in different environments undergo selective pressure. So they develop traits that are specific and allow them to survive within the unique environment that they grow in. Okay, so we're going to exploit this to understand adaptation to serpentine soil. Okay, and then my study system is Mimulus guttatus, these cute little guys here. And so the populations that I study are annuals. Not all of them are annual, but they're found broadly across Western North America. They've been a model species for a long time in ecological research. They have a lot of interesting stories to deal with speciation and hybridization, and they have this really high level of genetic diversity. Part of this is due to their outcrossing nature. But because of this high level of genetic diversity, they grow in this super wide variety of habitats. So we can find them all the way from sea level to 8,000 feet up in the Sierras. If you drive through the Central Valley in the spring, they are just in the ditches. They are a ditch plant. They grow on seaside cliffs here, and they deal with salt spray. There are populations that are found on copper mine tailings, so copper contaminated soil, and this is a side project that we work on. And then the work that I'm going to talk about today is how they survive growth on serpentine soil. And so what I'm interested in is understanding what genes or alleles confer this tolerance to serpentine soil. So we have no idea. So we got to take a pretty broad, multi-pronged approach to understand this. So I have some genomic data trying to figure out if there are specific alleles of altered proteins that are important for adaptation to this environment. transcriptomic data? Do we see different expression of genes that might also be involved in adaptation? And then the ultimate goal is to understand what traits do these genes control, because evolution doesn't act on genes, it acts on traits. Okay, so to introduce you to my populations, my off-serpentine population is called sod. It's here in purple, and it's from Napa County. And then as the crow flies about 30 miles north in Lake County, we have our serpentine soil, or our serpentine population called REM. And what's really important about these populations is that they live in quite similar environments. They experience quite similar heat and water availability. So we're really restricting the kinds of selective pressures that act on these populations and limiting that to the soil environment that they live in. Okay, and also there's serpentine all around us if you guys are interested in exploring it. Okay, so what does local adaptation look like in these populations? So I have both plants and soil from these two environments. So when we grow these plants on non-serpentine soil, there's actually not a negative impact of the serpentine adapted lines. So both of these plants look great. We can chart this for a long time. The serpentine, the REM plants grow bigger because they have larger seeds, and so they just have a little bit more oomph when they get started. But when they get to flowering stage, they're about the same size. There's no really difference in growth in this. Very different when we look at these plants on serpentine soil. So just to help you guys out a little bit, there's a tiny little seedling there. And so we can see quite a drastic difference, right? So these plants are doing quite fine on their serpentine soil that they're adapted to, and these guys are just like, they are on the struggle bus, okay? And so again, we can plot this out, and I just wanna say, funny undergrads are the best, because my undergrad that did all this quantification titled this spreadsheet Plant Murder Quantification, which was amazing to just get that spreadsheet shared with me. But what we see here is that, yes, they're not dying, but they're really not doing well at all. And we can carry this out, and these plants for about a month and a half will never get to the point of flowering. They just get like really purple, and they're suffering. So we can exploit this difference here to really understand what is going on to allow these plants to live in such a nutrient stressful environment. Oh, and here's the comparison. So the REM plants are still not doing as well in the serpentine soil just because it is a nutrient stressful environment. Okay, so some of the data that we have is from our collaborators at Duke. Jessica Selby did this work during her PhD, where she sequenced populations of on and off serpentine plants from all over California. And we find that there are about 70, depending on where we have our cutoff, highly divergent regions within the genome between serpentine and non-serpentine plants. And these peaks are actually really narrow. There's sometimes 1 to 4 genes, which is great. It gives us a good clue about what we think has been selected for. And within these peaks, there's about 160 genes that have non-synonymous SNPs. So that's one piece of evidence we have. Another is to look at transcriptome profiling. So I was really interested in understanding, yeah, okay, so we have these genes, but are they differentially expressed? Are they expressed at all? So to do this profiling, you can't grow plants in soil and then look at their roots. Not possible. So I had to develop this hydroponic system where I grew these plants in tubs, and the bottom of the tub we filled with soil from either site. We call this serpentine tea. So essentially you're soaking the water in that soil and extracting the nutrients that way. And then this way we can look at tissue specificity, because of course the roots are experiencing the stress in a very different way from the shoots. Okay, so just to confirm that this worked, I did some elemental profiling of the water, and it did, which was great. So, so we see that we have much higher levels of magnesium in the serpentine conditions and really low levels of calcium, and then also low levels of nitrogen, potassium, phosphorus, and sodium. Okay, so what does this data look like? It is a lot of data, and I'm gonna broadly go over it. So, okay, So this is just a heat map of root expressed genes, but there are thousands of differentially expressed genes when we look at both non-serpentine and serpentine conditions. And so what I just want to highlight here is that we have some genes that are only differentially expressed between our two parent plants in non-serpentine conditions, and we have a much smaller subset that are only differentially expressed in serpentine conditions, and then we just have this set that doesn't matter about the environment. They're just differentially expressed between these two lines. So Mimulus has a pretty nice genome, but it's not super well annotated, so doing Go analysis is not the best. So just digging into some of these genes, we see a lot of ion transporters that are differentially expressed, a lot of genes involved in nutrient metabolism, and then a lot of genes involved in cell wall biosynthesis. And so all of these kind of fit in with what we expect would happen. And then for the off-serpentine plants that are in the serpentine conditions, they are freaking out. And so there's a lot of genes involved in reactive oxygen species, a lot of genes involved in catabolic processes, such as protein degradation. Their shoots, they just shut off photosynthesis because they are not handling, like their ion homeostasis is so messed up. And they start to remobilize their central carbon metabolism. Okay, so we can zoom in a little bit and combine some of this data. So what I have highlighted here on the very far right over here is how many non-synonymous SNPs these genes have, what ion they are potentially either transporting or regulating, and then how they are differentially expressed in our data sets. And what I really want to highlight here is that it is not one nutrient, it is all of them, okay? And so this is a very complex environment, and we know this about nutrients, but usually we study them one by one. And so this gives us a really good platform to understand the interactions of all of these nutrients. Okay, so digging in even more, this is my only CRISPR slide. So we have a few early target genes. They are both ion transporters. So this 2-port channel 1 is a calcium vacuolar ion channel, and this potassium channel is also a target. So these genes have non-synonymous SNPs. They're differentially expressed. And then also this TPC1 gene has been implicated in adaptation to serpentine soil in Arabidopsis aeruginosa, but has no, there's been no like chemical or genetic proof of this. But these are our initial candidates. And I want to highlight that all of the initial work in designing these guides was performed by my former undergrad, Allison. And she also taught herself how to use AlphaFold. And so modeling this TPC1 dimer, what she was able to find is that in this EF hand where calcium is sensed, it seems like the serpentine allele has a smaller channel and maybe is more selective because this channel is actually non-selective between magnesium and calcium. So this might provide a reason for why we are seeing differences. We have a lot of things to verify with this, but it's a hypothesis. Okay, and I am gonna actually talk about some of this future work I'm gonna do because I'm really in the middle of this right now. So I have also all this transcriptomic data for reciprocal F1 hybrids. The hybrids are always, they are tolerant to serpentine. So it is a dominant phenotype, but we wanna understand the cis versus trans regulation of this. And so using the reciprocal F1 hybrid and allele-specific expression, I can dissect that out. I'm really interested in building networks to understand the nutrient response, both on and off serpentine in these two populations, and then understanding, and then hopefully when these mutants happen, I will be able to analyze them, and actually linking this to some traits. So it seems like the Casparian strip has some differences between these, and I'd like to do some root anatomy and understand developmental differences between that. But I hope that I've sold to you that this is a really interesting system to actually understand nutrient regulation, and to, we can maybe feedback nitrogen or feedback some nutrient and really dissect the interactions that are occurring within how nutrients are regulated across an entire plant. Okay, and with that, I'd like to thank the Blackman Lab, especially Allison, and my funding. And with that, I'll take any questions.
time for a couple of questions.
Hi, that was really cool. Do you see Mimulus on a bunch of other different kinds of soil? And do you think that the same genes might be involved in adaptation across soil types?
So I can answer this in the copper mine tailings situation. Not the same genes at all. Like those plants actually die on serpentine soil. Yes. I don't know that much about other soil types. I can learn more, but serpentine, because it's patchy and across California, I think it's where we would see it the most, at least in California.
Hi, I have a similar related question. I was wondering if, in addition to Mimulus, there are any other plant species that have similar adaptation to the, or yeah.
There are a lot. Yeah. There's a lot of Streptanthus species that are found on and off serpentine. I don't know if there are any that are both on and off, if they're the same kind of, we can compare between populations. But a lot of them are like pines and oaks. And so things that, you know, we can't really work as well with in our greenhouses, but there's a whole lot. But sometimes it's species, not populations. So it would be a little bit more of a broader comparison. Yeah.
Really nice talk. Serpentine soils are pretty abundant, and so is Mimulus. Do we have reason to believe that this was like a single, I guess, adaptation to serpentine soils, or has it happened a bunch?
Do you want to fund my K99? We think it has probably actually happened a bunch. Yeah, so it seems like there are 3 actually different ages of serpentine soils in California. So we have Sierra serpentine, we have coastal range serpentine, which is what I've been focusing on, and Klamath serpentine. And the Klamath serpentine is much older, and we see not always different, but some differences in the SNPs that have popped up from those populations. Yeah.
Hi, I'm an Arabidopsis person, so I'm kind of.
I used to be an Arabidopsis person, so don't worry.
So I just like very curious how you actually deliver your guides and how is like the process of transformation of these.
Oh, we do tissue culture.
Oh, you did tissue culture.
Yes. Yeah.
Okay.
Sarnae developed our protocol for tissue culture transformation.
Last, one more question.
Great talk, thanks so much. I'm curious about how the suite of these nutrients or nutrient deficiencies or mineral presence, it operates as a suite versus individual additive effects. And do you have a system to understand how to parse the suite versus the individual?
I would like to develop this. I mean, at this point, my hypothesis is that I can do this serpentine tea and just put in some nitrogen and then see what kind of differences we see. We don't know. I mean, for a very long time, it was thought that the magnesium-calcium ratio was the most important stress in this situation, but actually there's been more recent research that the lack of nitrogen has been a really important selective factor in like keeping plants that are not adapted off these soils, but it's all ecology research, and there's not been a lot of delving into like actual genes. So yeah, this is something I'm interested in doing in the future for sure. Yeah.
Thank you, Ali, for the nice talk.